Welcome everyone to the live session, and I'm going to turn it over to our committee member, Jason Delborn.
Thank you, Emily. Welcome to all those who are in the room as well as online. We're excited for today's session, which is titled Addressing Gaps in Environmental Safety and Security for Engineering Biology Research and Development. My name is Jason Delborn. I'm a faculty member at the University of Wisconsin-Madison, and I'm a member of the National Academy's Committee on Environmental Safety, Security, Evaluation on Engineering Biology. Thank you to the panelists who volunteered to come and join us today, those both who are in person as well as online, and also to all of you in the audience, both here and online. Today's panel will be hearing from 4 distinguished guests: Chris Wozniak, formerly from the Environmental Protection Agency; Kate Adamala from the University of Minnesota; Lema P. DeHaro from Merrick and Company; and finally, Vaughn Cooper from the University of Pittsburgh. So in our format, each panelist will make about 5 minutes of remarks, and then we'll have time for questions and answers at the end. And I'll turn it over now to Chris Wozniak. Thanks for coming to join us.
Well, thank you for the invitation, and it's good to see some familiar faces and meet a few new ones. So my talk will be particularly biased in the sense that, I've been in regulation for 22, 23 years now. And that's kind of what I've been doing since I retired as well, as consulting as a regulator. And this is probably my swan song, so thank you for this. This is probably the last talk I'll ever give. But be it as may, I'll.
That's what you said the last time.
I know. John, but basically, I think that one thing all of the techniques have in common, whether we're talking about cells, synthetic cells, traditional, transgenesis, etc., and just regular microbes used as biocontrol agents, they all require regulatory approval to move forward. And without it, unfortunately, a lot of that technology will never get to the public. The consumer will never see the benefits. And that's kind of what drives my path behind it, is that I think we should be way ahead of where we are now. So many things that have come past, my desk over the years, and things I see in the literature, and frustrated scientists telling me, yeah, I'd love to get this out there, but I don't have $500,000 to come to your agency. So I get that. Now, especially as I'm a consultant, I see the other side of the table and the costs that are involved. So as I mentioned, we're blessed with all these great technologies, but again, we need a functional science-based regulatory system. Now, there certainly are claims. Every agency will tell you that we're science-based on it, and there's some level of truth in that. There's also a lot that's left unsaid. A lot of things that are either politically motivated or just even done, I hate to say it, but out of ignorance. The pre-coded organisms and synthetic organisms that may use non-canonical amino acids and unusual nucleotides, even like that, are really going to throw regulatory system for a loop. Because a lot of things that people do are based on databases that are, looking at quote unquote normal proteins, normal sequences. And when you start throwing in, modified bases and amino acids that don't readily appear in nature, whether it's for allergenicity or toxicity or whatever the database you're using, you've got a problem. And I really don't have any idea how the regulatory system will handle something like that if and when it occurs. We've been told that it's coming for several years. I couldn't tell you where that is at. But the Coordinated Framework for Biotechnology, which was originally published back in the 80s, 84, and then really 86 was the first codification of it. A lot of people mistake it, think it's a regulation. There's no regulations involved. It's a policy document, a guidance document, if you will. But what it did, or what it should have done, what it was intended to do, was get the scientific community on the same page and say, okay, relatively speaking, genetically engineered organisms use the same principles and rules of physics, biology, and chemistry as the non-engineered ones. And pretty much everybody seemed to agree with it. Yeah, that makes sense. Okay. but then they turn around and create rules that are specific for those that are produced using recombinant DNA. And to me, that sends a message both to the developer, but also to the public writ large, that there's something different here, might be something risky here, you should really be paying attention to this. And this persists to this day. I'll just give a couple of examples here in a minute, but I think that's kind of the main driver for me, is that you still hear the rhetoric that we don't treat recombinant organisms differently. And then you look at the reality and say, really? So this disparate treatment, sorry, I should have been telling you that that's the slide. The disparate treatment of GE microbes, OPP, by the way, that's Office of Pesticide Programs. That's where I work predominantly. And OPPT is Office of Pollution Prevention and Toxics. They're the folks that predominantly use TSCA, Toxic Substances Control Act, as their authorizing statute. So in OPP, where I was, we could take an organism that was, I found a new species, a new fungus, kills an insect, bring it in, you can go out in the field up to 10 acres. You don't even have to talk to the agency. If you put it on a food crop and then sell it, now you have an issue, but just testing for proof of concept's sake. Now, if you introduce a marker gene using recombinant DNA, well, now you can't go out and do any size field test without permission. And again, not going into naming names, but a good colleague, he became a good colleague because he was so angry at the EPA. Basically, they ruined his field trials because his bacterium had a NPT2 antibiotic resistance gene in it. And it was, again, proof of concept. I just want to see if this thing will survive in a grapevine over the winter. They wouldn't allow that. And the reasoning is because he mentioned, ultimately, I want to put a toxin into that bacterium and use that to control a pest. Well, now you're a pesticidal intermediate. So you fall under the TSCA folks in OPPT. And, I try to get folks to explain this in a public forum. Explain to me why you had to torch the root zone. It sounds crazy. It reminds me back of the Moses Lake studies with the potatoes and the frost, the ice minus bacteria, everybody in the moon suits, At times it seems like, even though this started back in the 80s, I'm not sure that we've learned a lot, or at least we act like we have a lot. But again, under OPPT, if your organism only has basically rearrangements or elements from that species, you're already included in the TSCA inventory. You don't have to go for permission. Now, if it's between two genera, it's heterogeneric, now you do. But OPP doesn't see it that way. All the organisms, non-engineered, if they have an intent to use the pesticide, they require a permit. If you add in recombinant DNA, now you have an extra level. You have a biotech notification process. Well, this is the same agency. Two groups of people at the same agency. It's like, wow, was there no coffee much? So anyway, I'm sorry, I'm going on long. I just want to say that in terms of changes to something constructive, moving forward with this, I certainly don't have all the answers, but what I would say is simply the fact that there are decision makers in various agencies at the state level as well who don't grasp the importance of the technology, certainly don't understand it. Many of them who are making the decisions do not have significant scientific backgrounds. And to me, that's unconscionable. Basically, as I said, I think there's a lot of technology that we should have in use right now. And looking at, again, focus on the pesticidal microbes. I think we have 3 that are still in commercial use, since the 90s. We've had dozens of biotech notifications where these things get field tested on a small scale. Nothing commercial. Why is that? And on the OPPT side, I wish I could tell you what they're doing, because I found out when I asked them, they won't admit to almost anything. They won't tell you Well, I saw on your website, you say you have this yeast. Well, is that a Pischia? Is that a Saccharomyces? Can't tell you. Was it actually put in the field for use? Can't tell you that. Well, what was its intent? Sorry, can't tell you. Well, how about if I FOIA it? Won't do you any good. Well, isn't that special? I guess with that, I'll just say there are some other things a person could do, and I'm not the originator of this concept, but I liked it. Is there ought to be an appeals process. You're the developer, somebody asks you for a ton of this data, say, all right, explain to me why I need that earthworm study. Why do I need the soil study? Why do I need all this? And as of right now, there's relatively little impetus for the regulators to explain in detail why they need what they're asking for. And to me, that's just unacceptable. So with that, Josh, take any questions?
Thanks, Chris. And I think we're going to hear from all panelists and then have a discussion. So if people have questions, please remember those, and we'll refer those to Chris at the end. So next we have Kate Adamala from University of Minnesota, and she's appearing online. Welcome, Kate.
Welcome. Thanks for having me. So I'm a wet lab synthetic biologist who's been watching a little bit on the sidelines as we develop those technologies. And I'm sitting here thinking, why are we not regulated? Which is not a typical kind of a mindset. Obviously, we would love to run and do what we're doing, but it also is kind of a self-preservation instinct, seeing what's happening right now around us in our field and how there is this huge gap in regulation. I mean, I think everyone in this room agrees that synthetic biology is growing faster than the regulation that's supporting it. And Chris covered a lot of the points that I wanted to emphasize. So thank you for that. Thank you for kind of framing it. Some of the technologies that I would like to mention, that some of the things that Chris already mentioned too, are things like genetic code expansion. For example, it's not just the problem of how will proteins with novel non-canonical amino acids behave, but also the critical breaking of the link between the genetic sequence and the protein product. With the genetic code expansion, with recoding of sense codons, this so-called sense codon reassignment, it sounds like science fiction, but it actually is possible in the lab already. We're doing it. We can order a DNA sequence, and then using that sense codon reassignment, we can translate a protein out of it that is not predicted by any typical algorithm that uses the canonical genetic code. So, for example, one could imagine ordering a DNA sequence that's a complete garbage to all the screening algorithms, and then recode it so that it produces a toxin or something that's a select agent. That capacity already exists. It's still pretty specialized. It's not something that anyone can do, but it's possible. And for example, the sequence screening algorithms are not keeping up with that. Benchtop DNA synthesizers are also kind of breaking that framework. If you have the capacity to synthesize DNA on your bench, it's not just state actors or really well-resourced labs that have the capacity to synthesize their own DNA and bypass the screening algorithms. It's anyone who can buy a benchtop DNA synthesizer and jailbreak it to remove the safeguards, which is not that difficult. And then synthetic cell engineering, that's another layer of breaking all of the possible safeguards, because that's engineering organisms that didn't exist, that are not limited by the evolutionary kind of guardrails that are put on most normal living organisms. So how do we regulate that? How do we treat that even? And on a little bit simpler level, cell-free systems, which right now are very poorly regulated, are basically regulated based on the parent organism from which the cell-free system came from, are now, that technology enables things like synthesis of replication-competent viruses, which is great for research. It's great for biomedical applications. But for example, my E. coli system, I can take an E. coli, grind it up, and it's now completely dead. So it's not even BSL-1. And I can use that to make a virus that's capable of replication. And again, that's not really regulated. Another question is what happens once we start making organisms with artificial organelles? How will those organisms be classified? If I take, for example, an E. coli and engineer organelles into it to give it capacities that no bacteria by natural laws should ever have, what's going to be the status of that organism? That's something that's not really, the technology is on the horizon. It's happening in the lab. It will soon be available to anyone who can read papers. And we're not prepared for that. We're not prepared with the regulatory framework. And those regulations are not just needed for safeguarding the research, but also for applications. That field is driven by investment in practical applications in biomedical, in biotech, bioeconomy applications. And in order to scale that up, we need that regulatory framework that's actually easily applicable. That's something that Chris really summarized really well, is how difficult it is to get something analyzed, approved. That process has to be streamlined. And then last but not least, the citizen science movement. People literally cloning in their garages. That's great because it democratizes biology, it democratizes biochemistry, it democratizes that knowledge, but it also spreads the knowledge. It allows people with very little background to start doing what we used to think is only skills that are reserved for highly developed academic labs. That's already out there. There are literally thousands of those citizen science biolabs all over the world, in places with much less state level oversight as well. How do we safeguard that? These are the questions that I'm posing. I don't have answers to that, but I think we really need a more flexible framework. We need something that will be more responsive to those technologies as they arise. And one thing that I would like to end with, maybe as an opening for a discussion, is maybe we should regulate capabilities instead of specific technologies. So instead of having a regulation that says this DNA sequence or this particular type of an organism is regulated, maybe some regulation should mention the actual capabilities. So it doesn't matter what kind of an organism or technology is capable of doing that, but the X function is regulated rather than a actual physical system. And I realize it's a mess and it would be really hard to define, but we have to do something. So that's where I want to end. And that's my opening for a discussion, is how do we regulate that existing mess? Thank you.
Thank you so much. And do we have Lema Dehiro online? We do. Okay, excellent. I'll turn it over to you then for a 5-minute introduction.
Hello, everybody. Can you hear me?
Yes, we can hear you just fine.
Okay. Good morning, good afternoon, wherever you are. My name is Lema Dehiro. I am a senior scientist at a company called American Company. It's an engineering and design of labs company. So we specialize in designing high containment laboratories around the world. I am a scientist by training, molecular biologist, and then I made a switch to biosafety and biosecurity. And I think about this deeply all the time. I echo the concerns of the previous speakers, and I want to add more specifically the role of AI and how much AI is really going to propel the movement of the entire field forward at an exponential rate, which is already happening. And our regulators and our policies are inadequate to really appropriately mitigate the risk, the potential risk for these new technologies. And now we're also adding robotics to this. There have been humanoid robots. There have been several papers that have been published on automated labs that have been sort of proof of concept. Google DeepMind has published a paper last year, and there's been a few others more recent. I know it requires a lot of resources right now, but it's only a matter of time before these principles become more accessible to everyone and the cost is reduced. And again, we haven't thought about this. And if you apply all the previous risks that the previous speakers have mentioned, and you add AI and automated laboratories into the mix, the benefits are really incredible, but so are the risks. And I think what I would like to encourage this group to think about is that, in my opinion, we need to think of it as a one health approach or a one world approach to this problem. We can't think about it uniquely as a in the US. We need to think about these policies because we are all together on this planet, and what our neighbors do may affect us. What we do affects them, and so we need larger global governing bodies that hopefully will feel empowered to come up with guidance and policies that we all can follow. And I know there's many colleagues that are really looking for that guidance. And it should really come from us, from the experts, rather than policymakers that maybe don't have a strong background, so that the risks are not exaggerated, but just properly mitigated. And thank you for having me today.
Thank you so much. And finally, we'll turn it over to our fourth panelist for introductory remarks, Vaughn Cooper from University of Pittsburgh.
Good morning or afternoon. Thanks everyone for having me. I appreciate it. So I'm a professor in microbiology and molecular genetics at the University of Pittsburgh in the School of Medicine. And I've had the great fortune to study the process of microbial evolution in action for about 30 years in a wide range of taxa. My lab's been asking an array of questions, like fundamental ones about the nature of the evolutionary process, but more recently we've been focused on practical questions, like how opportunists can become better pathogens or how microbes can adapt to novel stresses. And these days with high throughput genome sequencing, this literally becomes an incredibly powerful kind of evolution-enabled genetic screen to see how microbes adapt to new conditions, no matter imagine whatever condition you have. And it's that sort of power and capacity that has gotten me interested in synthetic biology and adaptations by released organisms. And so when I brought that idea up and whether we can sort of translate our experiences to synthetic biology, there's sort of this major misconception that comes up often, maybe not by anybody who's tuned in here, but one I've heard is that when you put engineered microbes out in nature, they may be so engineered that they lack the ability to undergo horizontal gene transfer, maybe because they're recoded, or maybe because they're so foreign that they may not have like a normal kind of mating partner nearby. And what I want to say is that's not at all necessary for microbes of all kinds to survive and persist in the natural environment. So what we found over and over again is that microbial populations grown even in the strict absence of horizontal gene transfer are absolutely not limited for making major, achieving major innovations in their evolution. Rather, over and over again, adaptive novelty that enables their growth and persistence in a new environment is immediately within reach, even within just one mutation or just a few mutations. And that basically reflects billions of years of evolution for plasticity, meaning that these microbes have in their genomes seen an array of different environments and have memory of that. And so one or two mutations may allow them to unlock that memory within their genomes. And given that we do synthetic biology on the basis of existing genomes and existing platforms, it's likely that a lot of that capacity is already baked in. And so just to sort of summarize, in literally every bacterial evolution experiment we've conducted or collaborated with, we've seen them unlock abilities to grow in new environments like an antibiotic, a new host, alter nutrition, or so on. And so with all that as backdrop, I've really been wondering, what can we learn from environmental release examples where environmental release of microbes has been carefully studied using the mechanisms and processes that we in sort of the experimental evolution field do. And I've found very few examples in the literature. And this is a missed opportunity. This is a missed opportunity not just for the science that we want to better understand, but also to better understand and put boundaries on environmental risks. So as Chris noted, environmental release, it may be a permanent event, but that may not be a risk, or it may be. These are the evolution and action experiments I'd love to see well-described examples of, and they don't exist. And so I guess I'll just conclude by saying that more studies examining these possibilities under controlled conditions are really warranted. They're gonna help provide framework for understanding and generating policies for successful use of microbes in nature, and they'll teach us a great deal of how the engineered microbes we haven't dreamed of will adapt or perish in nature, and likewise also how to develop strategies to mitigate that. Thanks.
Thank you very much. So we will now enter our discussion part of this panel. So I welcome people on Zoom to put their questions in the chat. I'll monitor that, and people in the room, if you can raise your hands, I'll call on you to ask your question.
Am I allowed?
Yes, you are allowed to, especially because you're in the room. But we'll start with Felicia.
Well, thank you so much to our four speakers. We really appreciate your sharing your expertise. I'm Felicia Wu, John Hanna, Professor of Food Safety Toxicology and Risk Assessment at Michigan State University. And we were having a discussion amongst our National Academies Committee about narrowing the scope. And I cannot help thinking with some of the talks we have just heard, suddenly the scope has, again, really broadened. In particular, Vaughn, your talk about how some of these genetically engineered microbes released into the environment have the capability of evolving, and it may take only one or two mutations to unlock something that would allow them to thrive. And that's a little bit alarming in some ways, because that would imply that the risk assessment that needs to be done examining so many different possible scenarios. Can you think of any way to sort of bound this at all?
To bound it. I mean, I guess, part of this may reflect my ignorance in this field of sort of the interface of synthetic biology and environmental microbiology. But I would say that this is a classic kind of experiment that could be done to ask both about the timing, meaning the rate at which the adaptation would happen, and whether that adaptation is at all counterbalanced by the natural predators that the microbe may have in nature. So even though they may adapt in the absence of biotic stresses, in fact, most microbes die by viruses and protists any day, and that would be true for synthetic cells too. Where we can be, should sort of pay particular attention is when we think the way we're building these cells makes them more recalcitrant to that kind of ordinary predation. So I didn't really answer your question, but I think the absence of natural sources of death should be the first alarm.
Thank you.
Do other panelists want to comment on that question? Chris?
I can sense it sort of broaden it even further. I think Vaughn makes an excellent point that, and I meant to say this in one of my slides, that when we look at naturally occurring microbes, bacteriophage, insect and plant infectious viruses, fungi, and even some protozoans, we don't ask the question, how long do they persist in that field after they're used for whatever? And there's been a little bit of work where I look for, Bacillus thuringiensis israelensis. Does it stick around? You put it in the bird bath all the time, keep the mosquitoes down. I found one study, over in Switzerland, where somebody actually looked 20 years later. Oh yeah, it's still there. But nobody is worried about it. And so one of my points is simply the fact that yes, depending on what you're putting out there, if it keeps you up at night, you can't sleep because like, oh my God, I just approved the release of this gene off the select agents list, that's one thing, but I would argue that, as Juan mentioned it, in an environment where you have lots of naturally occurring competitors, predators, whatever you want to call them, parasites, it's not the norm. It's sort of contrary to the norm. But 10 years ago, I was doing some work with DARPA, reviewing some proposals and trying to explain the regulatory hurdles that they were facing. And I asked them if they would ask their people, they funded some great work, and I was able to see some of that firsthand. I said, I'm more interested in your failures. Tell me when your constructs fell apart. Because we had, for example, the first time, only time I'm aware, we approved a synthetic gene for use in the environment. It's in a plant infectious virus. And the argument is, well, that virus is going to, because of the metabolic cost of this gene or genes actually, it's going to fall apart. It'll go back to wild type. And we were kind of like, yeah, it kind of makes sense, but I've got zero data to back that up. So we asked these folks, if you get anything like that, and I know you do because you've already reported on it, but unfortunately, nothing ever came forward. I still think it's a very valid angle. The negative results could be just as impactful, especially for risk assessors, where they need to hang their hat on something. They can't just say, yeah, I think that's biologically plausible. I need data. And I think it'd be great if somebody, whether it's a mesocosm study or something larger, actually pursued that to see. There is some reference to that paper that I published with Jonathan Shemla and Chris Voigt a couple of months ago. He does address this to some degree. Jonathan did, in terms of putting microbes into environments where they have natural flora that's going to compete. Yeah, they tend to go down pretty quickly. And that's, again, not surprising. It doesn't mean it's absolute, but at least it's something.
Thank you.
And Chris, did you have a question you wanted to?
No, that's sort of the same thing. I was just wondering, if anybody else had really looked into that or is aware of anybody looking at these failed constructs. Because that might give you some hint of the design. If you want something that lasts weeks, not years, well, put it into this organism, and it will be organism dependent and likely sequenced. Put it out there. The one we worked on was a infectious virus that affected a tree. It's confidential. I won't get into what it is. A food crop tree. And sure enough, yeah, thing fell apart and went back to wild type. But we only knew that after the experiments were done, not before.
Thank you.
Cameron.
Thank you all for coming in to speak with us. Cameron Kim, Duke University. I think my question goes to Kate, but obviously for everybody here. Kate, you mentioned a lot of the interesting work with recoding and sort of refactoring of these genomes. And one of the elements of our statement task is to look at these synthetic cells or the use of other types of cell-free types of systems in order to generate whatever, right? And so I think kind of from a scoping, understanding kind of where we are in the field right now, you made some comments around you can order a DNA sequence to translate a non-predictable protein. So I'm kind of curious sort of what, maybe it's a question of what examples exist if you have any to comment on, but also what then needs to happen, especially when we get to say de novo protein designs, where again, we don't have any idea about how those proteins that, are being computationally designed work. And given some of the concerns with these types of synthetic systems that we may or may not have any idea how they're producing, what they're producing, what might you recommend in terms of maybe some of the application regulatory space that you mentioned? What should we be thinking of or prioritizing in these types of frameworks from applications that may be the most pressing or the most concerning for this committee?
In terms of applications, it's possible to recode, 4 or 5 sense codons, which is enough to completely obfuscate a sequence. We've actually done some work with someone who runs those algorithms for Twist, which is one of the big DNA synthesis companies, and we gave them our sequences. They pretended that they were benign reporter proteins, but they treated it as a select agent and tried to screen it as what if someone put that order in, and we were able to basically put those orders in. If that was an actual select agent sequence, we would be able to order it and not be detected. So that's the capacity. It's possible to fool the sequence screening algorithms enough to order whatever select agent you want. It takes some assembly. You cannot order a complete genome. You have to splice it into small pieces, but it's basically trivial to assemble it back into a plasmid. In terms of what could be done, maybe I underappreciate the field, but I think there is enough perfectly fine, perfectly deadly natural pathogens that right now I'm not terribly worried about the completely engineered proteins, because what we worry about is not just how harmful the protein is, but how easy it is to weaponize it, how easy it is to spread it by accident or intentionally. And the de novo protein design helps you design, for example, perfect receptors, potentially perfect new toxins, but they will be very difficult to spread. And there is a lot of viruses that nature gives us that are wonderful potential weapons, and there's no need to make new ones. If you want to do harm, nature gives you plenty. So but that's actually good, because that means what we have to worry about is screening against existing known pathogens. And the good thing about them is that they're known. Right now, we're screening against their genomes, but we also know the frequency of amino acids in their sequences, and that's something we can screen against. We're not doing that right now because the algorithms are using DNA alignment. So basically, they screen against a known DNA sequence, but it would not be computationally prohibitive to screen against amino acid sequence in known select agents. And that would be a relatively low-hanging fruit to safeguard against that particular type of genetic code expansion. That's one actionable item that I can talk about. I have no idea what to do if someone completely designs a de novo virus. I guess we just go and die, because there's nothing to screen against that if we don't know it. So I would rather focus on things that are doable, and screening against known doable pathogens using amino acid frequency rather than DNA frequency is one thing that we can already do.
I would add that I think we also need to talk about the loophole in that, which is the benchtop synthesizers. And I remember last year I spoke at an event at an institution that I won't mention, and there was a scientist there that works with pathogens for legitimate reasons, and they were frustrated with the length of time that it takes them to order genes. So they just went ahead and ordered their own synthesizer for legitimate reasons. But these synthesizers are now accessible in terms of cost and easily obtainable. And someone with nefarious intents could as easily obtain them just as a legitimate scientist with a legitimate purpose. And I don't know that these instruments are being properly regulated worldwide. In fact, I don't think so. So when we think about this, I know there's a lot of focus on gene sequencing screening, but also we need to think about this loophole. And then the other thing I want to bring up is that, remember, the gene sequencing screening, the gene sequencing companies have a consortium, and they do this voluntarily. They're not required to do this. And there could be gene sequencing companies that choose not to participate. They don't need to. They're not being forced to. They're not required. And that's a gap. That's a loophole that needs to be closed globally, not just nationally. Because if we do it here, but we don't do it elsewhere, then that becomes the loophole that we'll be constantly chasing.
Thank you all. So I'll ask one. I think, looking across the different presentations, there's a lot of agreement, but there is this tension between a kind of regulatory burden that has hindered innovation and also a sense that new things are coming that the regulatory system has no idea how to even recognize or deal with. And so in some ways that's a tension, but it's also an opportunity to be able to do something different with this next wave of innovation that provides the kind of security and safety that we're looking for without the kind of burden that has hampered innovation. So are there ways that you see ways forward in terms of frameworks, ways of thinking, or particular strategies that we could use to create governance over these emerging technologies that might really challenge our current system without hindering innovation?
Yeah, man.
Sure, yeah.
Well, I mean, one of the things I think that is definitely needed, as we've alluded to, is the fact that a lot of this is very novel new technology. And I don't know what it's like anymore because I haven't worked in grants for 20 some years, but I can bet it's difficult to come up with funding to do the types of mesocosm studies that would really be informative in terms of survival of a microbe. I mentioned in my slide that persistence does not equate with risk. And I believe that to be the case. Doesn't mean it can't in certain circumstances. And so the only way to do that without just putting it out there, let's see what happens, is to do some sort of a controlled, contained type experiment. And people have talked about that. I mean, especially with the engineered mosquitoes. There's a group out there, APHIS group in Fort Collins, I think. Anyway, people are thinking along those lines, but I'm not sure how much that's been done with microbes, particularly with, if you want to get into like synthetic cells or things like that. I'm not really sure. Does anyone know it? Are there active projects with things like that? Because that, to me, again, going to a regulatory agency and saying, here's what we got. Yeah, we don't think it's going to persist and everything, and that doesn't cut it. You're going to need some sort of an experimental design that at least convinces the regulators at one or more agencies that this is plausible and not going to cause irreparable harm to the environment or human health.
I think it's just a really quick follow-up on that is if for research grants to do that kind of testing, is there a field trial, mesocosm style approval process in place currently?
Or depending on what organism you're talking about. So certain organisms might be regulated by APHIS, USDA, and certain ones by FDA and certain ones by EPA. So I'm not aware of any specific, something they call this is a mesocosm process, but I do know that the Office of Research and Development at EPA, at one time I visited, I actually reviewed the lab on Corvallis when I worked for USDA, and they had mesocosms set up, with X number of different plant species, things to study, but I can't say that I'm aware of any regulatory agency utilizing the data that came from there for a specific purpose. Certainly with the mosquitoes, and that's something that I draw a blank out researcher's name. But anyway, we met before when I worked on the Oxytec mosquitoes, and I hope that work is still going on. I don't know.
Just a quick comment. The only examples that came to mind that I could recover when I was thinking about my remarks for today were in highly controlled studies of microbes adapting to colonize plants, both here in the States and in Europe, in greenhouses, basically.
I'll make one more comment. I sort of alluded to it earlier. We did have a couple of scientists from Beltsville years ago contact us, and they were looking at E. coli as a human pathogen, but curious at a mock-up system they had watching tomato plants and tomatoes and things out in the field. They had this E. coli, called a lab strain, a K12 derivative with a marker gene. And then when I was like, well, what do we got to do to get approval to do this? And it was actually, it was a challenge for a lot of colleagues. Like, I don't know that anybody, because you're not making a pesticidal claim. So who would care to regulate this? And we finally just told them, just go ahead, just follow good basic biosafety level 2 protocols and do that. But it does raise the issue, because if you went out in the field like that and you said, my ultimate aim is to take that E. coli and then I'm going to further transform it and I'm going to add this gene. Oh, maybe you got a pesticidal intermediate. You better go talk to the Office of Pollution Prevention and Toxics. And that's something you have to be wary of, and most people, scientists, wouldn't know that. It's, they say, don't say too much to an insurance salesman. Just, if the lawyer asks you if you want to watch, say yes or no. Don't tell them the time.
Richard and then India.
So I wanted to ask, maybe this is Chris and Layla, a little bit about biosafety of sort of non-living things, right? So I'm just going to put DNA out in the environment. Maybe I'm going to use a gene gun. Maybe this is something that's a virus, but I'm not actually putting it inside a living organism, right? It's just the chemical DNA, right? That's sort of out there, which could be from a decayed living organism, but I want to do it where it's not in a living organism. How does that get regulated or analyzed for biosafety?
I'm not aware of anything that's present. Certainly for double-stranded RNA, there is a process.
Right.
EPA has registered, actually the last product I worked on, I think it's Greenlight Biosciences was doing some work synthesizing double-stranded RNAs for insect control.
Okay.
And I think there's also a product in Canada, the Colorado potato beetle, that is approved. So they'll look at it does not have to be a living microbe. Because obviously EPA regulates thousands of chemicals, both under TSCA and under different pesticide statute. But DNA, if you just say, well, put DNA out there, I'm going to look for some sort of horizontal gene transfer and see if something mutates, right? I mean, without a pesticidal intent, I'm not sure who would regulate that. I mean, if you brought the DNA in, this is real hypothetical, but if you brought the DNA in from Canada.
Yeah.
Now, APHIS, plant protection and quarantine, I look at that and say, well, that's imported and that's part of a living organism. So we may need to look at that. But, I'm not aware of anybody doing that kind of thing.
Okay. Well, Layla, do you know anything on the just biosafety side of things, maybe?
So in biosafety, there's no unified guideline in terms of controlling release of DNA. Once an organism is inactivated, that's the end of biosafety, according to the BMBL. Sometimes at the more local level, if you're an institution and you have an IBC committee, maybe if someone has a concern, usually most concerns that are brought up are not from the safety angle, but more from the experimental integrity concern. The scientists may be concerned about cross-contaminating their samples with different DNA, or putting in jeopardy cell lines or animal strains or plants, more so than actual concern for safety. My personal opinion is that is a gap that should be addressed, and not just for DNA, but we also have prions, which do not fit any definition of a living being, but yet they can be highly concerning from a biosafety and biosecurity point of view. And again, prions are not necessarily regulated from the biosafety point of view. They're more at a local level from actual concerns from the experts that may be looking at specific project or experiment, more so than a unified, universal set of guidelines or rules or policies that would cover everyone. As our colleague mentioned, he's looking at it from the EPA angle. Usually when we're looking at biosafety, we're not thinking about the EPA at all, right? And these groups are not talking to each other, which is also a gap. We need to be talking to across agencies, across groups, that goes back to what I mentioned earlier about the one health or the one world view, because the USDA thinks, well, I'm just the USDA. I don't care about animal pathogens, or I don't care about human pathogens. The CDC may be mainly concerned with human pathogens, except pathogens that may be vectors. And then both groups are concerned. But if it's just a plant pathogen, usually one agency doesn't care while another agency does care. And then we fall into these gaps. So, and I think DNA is one of them. I have personally served on IBC committees where there were concerns of releasing DNA material in a greenhouse. So that IBC committee specifically asked the scientists to put protocols in place to basically destroy the DNA before releasing the waste as biohazardous waste, right? But that was a unique scenario, and it was above and beyond overabundance of care and caution that was absolutely not required by anyone beyond that group of people.
Great, thanks.
I think it brings up a really good point in that, and not criticizing you, but to be fair, I wouldn't say that the agencies don't care.
Yeah.
What it comes down to is the statute authorizes you to do X, not Y. Even though you think Y is a good thing, I got chastised. For those of you who remember Hugh Iltis, speaking of Madison, quite the character, and he's like, Chris, you need to go down to the border and work on a gene flow on corn in Mexico. I'm like, no, I can't. And that's the way it is with these, APHIS is looking at plant pests. They also have Animal Health Protection Act. And believe it or not, I was working with them with a client last year, and they even look at like protein hydrolysates from animals that's coming in. So it's what their statute tells them they need to do. But it also raises the point of, from more an EPA perspective, do you have to regulate everything that you can? And to me, that is still an open question. You may have noticed that APHIS was moving with the secure rule, moving towards, they had a roadmap plan for GE bacteria, an off-ramp, if you will. And they also had already, I want to say exempted isn't the proper term, but basically familiar plants, another Roundup ready corn plant and another glufosinate ready or tolerant soybean. And I thought this makes sense. They're not wasting resources, and we've seen this 20 times. We're good. And then the motion got vacated in court. So that's kind of where things end up, and that's why, especially in the current climate, with a lot of people working with reduced staff, to dedicate a group, we really need new regs and maybe even a new statute, cap oil. But to sequester those people and say, look, for the next 2-3 years, you team of 8, you work on this, and that's all you're going to do. We want this, and agencies are just really reluctant to do that because they've gotten burned before. We worked on a PIP rule years ago, 8 or 9 people for 5 years, and it went up in smoke. Got nothing. So those managers are not going to make that mistake a second time.
So we have 2 questions, India in the room, and then we have a question online from Melinda that I'd like to give her a chance to read as well.
Just leads to so many more questions. I was wondering, and you mentioned, in terms of frameworks and the idea of needing negative data or other kinds of just data. I'm wondering what kind of framework is there, or is there anything relevant that we should be thinking about in terms of invasive species or novel things that are, because, in some ways what we're talking about, just because it's synthetic or otherwise, it's really potentially, it's like a non-native species that's introduced into the environment. And so whether that's, something from Tennessee into Hawaii or something else. So what is there that's like that we should be thinking, is there anything like that that's useful? And is there research being done that would be useful in this context, but is just somehow siloed, and so those things aren't connected that we could kind of point to?
I think there's opportunity for collaboration. There's a group in NC State, they're APHIS, I think they were called, say, CFIS or something like that. they did invasive weeds, things like that. And I've always felt that, if you really want to talk risk assessment, let's talk invasive species.
Right, yeah.
Wasting our time on another metarhizium or a BT or whatever. No, let's talk about, that fish that just came up the canal in Florida. But I'm not aware there was a national, was it Invasive Species Council, Department of Interior. And I used to be part of that group. Again, I apologize. The guy's name was Stosh, and I can't remember his last name. I don't know if that still goes on or not, but they had representatives from multiple agencies. And that's the kind of thing where I think, particularly like ARS, for example, I used to work for ARS. They have labs all over the world. They're bringing in species. I'm like, that's the kind of risk assessment I want no part of. Because to me, that's really difficult work to, look at brought a hawk moth in to control leafy spurge. What's it going to do when it's done? Well, don't ask me. So I don't know what it takes. More funding, somebody at Interior or ARS or somebody to say, let's talk, because you're absolutely right. The invasive aspects of a synthetic cell or something could be mirrored by naturally occurring species that are just foreign.
Melinda, would you like to unmute and ask your question?
Yeah, sure. So this is in relation to Lema's comment about the loopholes and then also Chris's comments about regulations. So we know realistically, it's going to be really difficult to get any new regulations passed, much less a law out of Congress in this current environment. So, and even without those limitations, I think regulations are always a bit reactive and a little bit behind. So I was asking about the role of researchers and developers in governance of their own work and encouraging and promoting biosafety. What safeguards do you have within your own community? Are there ongoing efforts within your community to pay attention to the safety focus issues?
Okay, would you like to go first, or do you want me to go?
I can just briefly say one example that is the community coming together to regulate is a synthetic cell engineering community recently came together and spoke up against one type of synthetic cell engineering. The organism made of molecules of opposite stereochemistry. And we said there is no way to contain it, so it should not be made. And this thing doesn't exist yet, so you cannot technically regulate it because it's not a thing, but we know what foundational technologies are needed to make it, and we said we're not going to do it. We're not going to develop those technologies that would eventually enable it. So that's kind of a bottom-up self-regulation. We'll see if it takes, but that we're trying.
So I agree that there are a lot of initiatives from different groups of scientists and subspecialties that are trying to self-regulate and put guidelines in place for themselves. And I agree that regulatory agencies are very slow moving and in a constant state of catching up. So yes, I agree that we need to be proactive and maybe create our own guidelines. But then that requires agreement. And there are conferences, there are biosafety conferences and biosecurity conferences that experts attend. But for example, those biosafety and biosecurity conferences are usually attended by regulatory compliance people that maybe support staff at your university, or biosafety officers on boots on the ground. So they know a lot about regulations. They know a lot about the NIH recombinant DNA guidelines. They know about the Select Agents program. If you happen to have one in your institution, they know about the BMBL, but they may not be scientists. They may not have a practicing scientist background. So we need the scientists to be present at these conferences too, even though maybe they don't carry the gravitas and that more scientific conferences would. And vice versa, we need to create pipelines to create compliance officials and biosafety and biosecurity professionals that do have a background in science, that do have a better understanding of the experiments. But in addition to that, I would say this would require agreement between everyone. I have been a biosafety professional, boots on the ground, and I do have a science background. And sometimes there's a lot of pushback from the scientists because they think that when we ask you to do a risk assessment, we're just delaying your progress, right? We're just creating obstacles in your way that are delaying the progress of your research. And you could only go so far, but we're slowing you down, right? And many times the scientists will push back and say, we'll make a recommendation, and they'll say, well, why do I have to do that? Who's making me do that? Is that, and the NIH guidelines are a requirement if you have NIH funding. But if you do not have NIH funding, if you have private funding, if you're a private corporation, you don't have to follow the NIH guidelines. The NIH cannot force you to, right? The BMBL, which guides biosafety, it's a guidance. It's not even required. You don't have to follow it at all. The only thing you may be required to follow is the federal select agent rules if you happen to work with select agents, because otherwise they'll remove your license and you can't have the agents. But yeah, a lot of these things are voluntary, but there needs to be the goodwill from everyone saying, we're all on the same boat here. We're all trying to achieve progress of research without stifling the progress, while simultaneously trying to keep the risk at an acceptable level. I always say the risk will never be zero, right? If we engineer a new microorganism with the intent of maybe breaking down microplastics that are contaminating our planet and our internal organs, and someone says, well, there may be a risk of gene transfer, or there may be another risk, we need to agree, will the benefits of this new microorganism outweigh the risks? And we have to be willing to say yes or no, right, after looking at some facts. But we may also have limited data, and we have to be willing to proceed with limited data and say, yes, we move forward, we agree, right? And we'll proceed cautiously. But there needs to be agreement from everyone. And I think that's really hard to achieve as well.
Thank you. And on that note, we will bring this session to a close. We're at the hour. So I'd just like to thank Drs. Wozniak, Adamala, DeHaro, and Cooper for joining us and providing us with some really excellent perspectives and insights and case studies for us to think about as a committee. And thank you for all of those who signed in to join us for this discussion. Thank you.
Thank you very much.
Thank you for having us.
Great. Thank you, everyone, for joining us. For this open session, I will be turning it over to our committee member, India, who will be moderating. Thank you, India.
Thank you. Welcome, everyone. Really excited. We're delighted to have joining us today members from our staff from the National Security Commission on Emerging Biotechnology to share with us about their recent report. My name is India Huff-Barnard. I'm the Executive Director for Engineering Biology Research Consortium, or EBRC. And I'm a member of this committee, the National Academy's Committee on Environmental Safety, Security, Evaluation on Engineering Biology. So I really want to thank those of you from the NSCEB who are joining us today, and also for those of you who are joining us online. So we're going to have I think, great discussion. Today, we're going to hear from Caitlin Fraser, who's the Executive Director of the NSCEB, Samwise Evans, who's a Senior Policy Advisor, and Stephen Moss, who's also a Senior Policy Advisor. And they're going to be giving us a brief on the report, charting the future of biotechnology. And then we'll have a focus that's discussed, a discussion that's focused on aspects of it that's particular to our study and most relevant to safety, security, and environmental considerations for the future of engineering biology research and development. So now I'll turn it over to Caitlin and to the NSCEB.
Great. Thank you, India, and thank you for the opportunity to be with you today. I'm really excited to
share some greater detail about elements of our report that we think are relevant to your work. Again, I'm Caitlin Fraser. I'm the Executive Director of the Commission staff. I'll just talk for a couple of minutes about, to sort of orient you to our work and our scope. And then I'll ask Stephen and Sam to carry most of the water when it comes to the detailed recommendations that we want to talk through with you today. The National Security Commission on Emerging Biotechnology was created by Congress about 2 years ago. Congress has a practice of creating these legislative advisory commissions, particularly on emerging technology issue sets, anticipating that it's going to need to legislate. And over the last 2 years, our commissioners, who are a bipartisan, bicameral group, have been working, with us, with the staff, to develop the report that we made public about a week and a half ago. You'll see, we verbatim our charge here, our mandate here, but in the statute that created the commission, Congress gave us several prompts to look at very specifically, and then a fairly wide latitude to take the work in any direction that the commissioners saw fit. And so we made a few early scoping decisions that I want to just mention here, because I think they're relevant to some of the decisions that we made about recommendations. The first is to define national security quite broadly. We had the opportunity to define it quite narrowly in terms of defense and intelligence applications of this powerful technology, but the commissioners felt strongly that that would be, far too narrow and really miss the forest for the trees. So they talk about national security in the context of economic strength, economic security, food security and agriculture security, energy security, supply chain resilience, and then, yes, of course, the defense and intelligence applications of technology. Another significant scoping decision is to really lean into the word emerging in our title and to focus our energies on the newer tools that have been developed in the biotechnology sector over the last decade or so and what's coming. We did not look at things like pandemic preparedness or traditional vaccine technology or small molecule drugs, and all of that was quite intentional to be forward, as forward-looking as possible. As I mentioned, our commissioners are a bipartisan and bicameral group. We feel quite lucky to have Senator Todd Young at the helm as our chairman and Dr. Michelle Rozo as our vice chair. And as you can see, we have 2 sitting senators and 2 sitting members of Congress from both parties on the commission. The remainder are a cross-section of folks from academia, industry, and philanthropy, all of whom, and many with former government experience, all of whom have different aspects to work. Slide, please. As I mentioned, we got started about 2 years ago. We made our final report public just about 10 days ago. And the commission's authority extends through the end of next year. So we're moving into kind of phase 2 of this, phase 2, phase 3, depends on how you count, next phase of our work, which will be twofold. One, working alongside stakeholders and advocates to move these ideas from ideas to implementation. And I know that's not the purpose of this meeting today, but if in your non-academies jobs or lives, you'd like to be a part of that, we're very much encouraging and inviting folks to help us push these things forward with policymakers, both on the Hill and in the executive branch. And also to deepen some of the work that we started in the report, but knew that we either didn't have the bandwidth or the time in the moment to go as deep as we'd like to. And so there's some places, in particular on regulations, where which Stephen's going to cover what we said in the report, but we do anticipate doing a lot more work in that space over the coming year. All right. You can keep going. I'm going to breeze through our findings. I should have said at the outset that we're happy to take questions as we go. There'll be some sort of natural pause points, you know, in our transition between us that will invite questions, but please do, you know, raise your hand if I don't cover something as deeply as you'd like. Our key findings boil down to 5 things. The first is that while the United States has enjoyed decades of leadership in biotechnology, our risk, the risk is quite high that we are about to lose that edge to China. We call for policymakers to take action on the 50 or so recommendations in our report in the next 3 years. And we think that that sense of urgency and immediacy is quite critical in that, you know, just a year ago, when the commission put out its interim report, we said the United States has enjoyed decades of leadership, but China is sprinting to close the gap. That's the language that we used a year ago. Now, what we have said is that there are clear metrics against which China has closed that gap and is starting to exceed the United States. Not in every metric, certainly, but in things like manufacturing, capacity, and talent, and even frankly, in terms of the cutting edge research, we are starting to see that gap close quite uncomfortably. Our commissioners have been talking about, you know, in other technology sectors, China is known as a fast follower, right? A technology is developed and commercialized first in the United States or in another Western country, and China has proven quite good at scaling that technology more cheaply and quickly than other places can, and beating out competitors using this kind of brute force economic playbook that we talked about in our report. That's happening in biotechnology, certainly, but we are also starting to see that this is no longer just fast follow, that we are being out innovated in some areas by already the United States is like I said, uncomfortably dependent on China for key nodes of our supply chain, both the biotechnology supply chain, but of course other critical and emerging technology supply chains. The rare earth and critical material ground is pretty emblematic of the dynamic here. Biotechnology affords us the opportunity to, by using biotechnology, we can replace dependencies on China for key nodes of our supply chain. And we can also lessen the potential for China to weaponize those dependencies or geopolitical leverage or economic leverage should they want to be able to do that. Next slide. By reshoring a lot of this capability, by using biotechnology, biomanufacturing to make things here in the United States for which we are currently dependent on China or countries that are too vulnerable to China, we can create enormous economic opportunity all over the country. One of our key messages, particularly to our congressional audience, has been that while biotechnology, you know, I think in most people's minds, biotech is sort of the domain of like Boston and Silicon Valley. Of course, yes, it is, but we are seeing already, and the commission anticipates enormous growth, particularly in biomanufacturing all over the country. Indiana, which is Senator Young's home state, is a perfect example of this. It's the headquarters for Eli Lilly, and they have, you know, a really significant R&D presence there. But they are also, Indiana is home to a brand new multi-million-dollar bioindustrial biomanufacturing facility. Richmond, Indiana, I think is a place that many people would think about as a biotech hub. But they are creating, you know, hundreds of great biomanufacturing jobs in this state, whether that is, you know, R&D, you know, jobs in a lab, or more like manufacturing jobs that don't necessarily require an advanced degree, and everything in between. Next slide. I spoke in kind of my scope note about you know, the way that we looked at national security very broadly. We did, of course, with our origins in the Armed Services Committee and our mandate from Congress, have a deep area of focus on biotechnology's applications for defense. The commission decided, after looking at kind of this very broad set of the applications of biotech for defense conventional military capabilities, that biotech has the potential to be as transformative for our conventional military capabilities as aviation was at the beginning of the last century. So if you think about, when the Wright brothers were the first to flight, I don't think anybody appreciated that we would use airplanes to move things around, move people around, to do surveillance and reconnaissance, to do, military power projection. And, certainly aviation wasn't appreciated as that kind of general purpose transformative technology at the time that it was first developed. When we look at some of the breadth of applications of biotech, we started to think about it as truly a general purpose technology for defense, an enabler of other conventional military capabilities. So things like using biomanufactured coatings for airplanes or ships that perform better than the coatings that we have now, or fuels, or the ability to create tabletop bioreactors where service members can make what they need, where they need it, when they need it, and on demand, pretty far forward, lessening the logistical tail that has to follow a forward deployed unit. DoD is already leading quite a bit in this space, but we think that there is a great deal more potential to explore and to put in our warfighters' hands than we are currently doing today. Next slide. And then finally, and this kind of takes us to our recommendations. What is the theory of the case for the Commission's recommendations? It is a couple of things. We propose a 2-track strategy designed to both run faster than China and slow China down. We know that the vast majority of the cutting edge R&D is happening outside of government, often with government help or direction or funding, but not necessarily in government labs or government programs. So how can government play the role of being a better partner and a better customer for the innovations that are coming out of industry and academia? Like I said, we make about 50 recommendations in our report, in our unclassified report. They are roughly evenly divided between, just to use these terms, promote and protect. We're going to talk about a handful of them now. And what I just want to emphasize here is that we see that promote and protect as pretty inextricably linked. The ability for the United States to be at the leading edge of the technology, to be the place that the best biotechnology research, development, and commercialization happens, putting these products in the hands of consumers, positions us to be in a position of strength with respect to the protect measures. So the ability to lead the international community in development of norms and standards for the prevention of misuse of this really powerful technology is in some ways in a lot of ways driven by our ability to lead in the promote sector. Let me leave it there. I'm happy to take any questions at this juncture, and then otherwise would love to turn it over to Stephen to get started talking about a couple of the recommendations that we thought were most relevant to the scope of your study.
And I'll just note for those online, our committee, if you want to add questions to the chat as well, and then I can call on you, or just in case I don't see your hand. But yeah.
Stephen?
Oh, great. Great. Well, thank you. Thank you very much, Galen, and thanks to all of you. It's a real pleasure to get to present to this group. My name is Stephen Moss. I'm a senior policy advisor with the Commission, as India said, focusing mostly on the pillar of our work related to out-innovating our strategic competitors. And I've worked in a couple other areas as well. In my previous job, I was actually at the National Academies as a staff member, leading committees like these. So it's really interesting to be kind of on the other side of the table. So I appreciate the opportunity to serve in that capacity to help all of you as you continue the important statement of task that you have that's been put forward for all of you to pursue. So that being said, one of the things that we wanted to do in presenting to all of you is to make sure that our presentation was tailored to the work that you all are responsible for pursuing within your charge. And so because of that, we've emphasized a couple of our pillars, and we're going to go over those as we move forward. So what you see here is kind of all of our policy objectives within the report laid out. This includes, biotechnology across the US government, mobilizing the private sector, maximizing biotech for defense, out innovating our adversaries, securing biotech workforce of the future, and then of course, working with our allies and partners. And so for this particular presentation, we'll be focusing on recommendations that fit within the mobilizing of the private sector and out innovating our adversaries. And admittedly, we're going to kind of mix the way that we would normally present these, just for the sake of kind of going through particular components of each of these, of all of these recommendations. So to start, we're going to talk a little bit about one of the specific recommendations around grand research challenges. Caitlin already mentioned that and did a good job of kind of noting that the promote and protect side of things are pretty well balanced in the report. We're going to focus on one specific innovation R&D or two specific R&D recommendations that we have in the report. But I'm going to walk through a little bit of the background on those and why we think it's critical for you all. And I want to reemphasize the point of, promoting innovation and promoting R&D being very important when it comes to all these aspects of safety and security that you all are considering. Of course, it's very difficult to understand and implement any protection measures if you don't know the technology or if the technology isn't advancing. So that's one key component of what we found through our work and is sort of driving these particular sets of recommendations. So within the pillar of out innovating our strategic competitors overall, we have several different recommendations. This pillar starts by talking a lot about biological data, especially curating and collecting biological data. We also talk about protecting biological data under this component. And then it sort of moves to talk a little bit more about infrastructure and also resources needed for research and sort of the framing around how we think about research and innovation. Within all of those, I want to talk specifically, as I said, about the Grand Research Challenge. There are two that we have that are outlined here, but we felt that the reason that this landed as kind of a Grand Research Challenge is because we at the Commission felt very strongly about the need to keep innovation in the United States, or to keep innovation strong in the United States, and to ensure that we have sort of a North Star for our researchers to attract talent, to bring in the best innovation, and to continue moving forward on biotechnology. And so with all of that in mind, the way that we felt we could do this best was to actually think through what are some more specific goals that researchers could gear their work toward, where they could start to think through and be rewarded for accomplishing really amazing and seemingly unachievable feats in the field of biotechnology and engineering biology. You'll notice the recommendations up here outline two of these, but these are sort of the umbrella grand challenges as I've kind of been referring to them. So, Congress should initiate a grand challenge around focus on making biology predictably engineerable, or biotechnology predictably engineerable. It's very broad. There's quite a lot more that we're going to dig into on that specific area, but it covers a lot of this idea that other fields of engineering have the ability to routinely make end products, and in biology, it's much more of a challenge at this juncture. So how do we move the needle and continue toward that pathway of being able to actually produce end products more routinely? The second one here is related to scale-up. Scale-up is also critically important. It's something that we haven't emphasized in the past and something that we should emphasize in the future. Making it predictable rapid and cost competitive, as it states in the recommendation here. And we'll dive into some of the areas that are most relevant for all of you as we kind of move forward here. So I want to start with the first grand research challenge related to making biology more predictably engineerable. I'll admit that this is the first time post-report launch that I've gotten to dive into the technical details here. So it's going to be fun for all of us. I do think that it reflects a lot of kind of the current state of science and the way things have been moving for a while. We outlined component and keystone challenges, and I'll explain a little bit about what that means. With these component challenges, we're thinking a lot. These are still very broad questions. These are still challenges that have a lot of different parts, but these are areas where we start to map onto the engineering paradigm of modeling, making, and measuring. And so finding different ways to do all of these different to pursue all these different areas and be effective in these specific component challenges. And then in the second part, I have these keystone challenges. In the report and in the appendices of the report where we go through all the keystone challenges, there are actually 18 of them listed. All 18 go through about 4 different categories of foundational research, advanced measurements, experimental tools, and computational models. And then the ones that I have listed here map a little bit more closely to what you, as in your committee capacity for the National Academies, are responsible for, related to minimally synthetic cells, mapping and measuring of molecular interactions, and of course organoids, organs on a chip. So as you pursue kind of your own statement of task related to risk and related to safety, security, environmental release for these different components, it's important to think through kind of what is the most advanced research, where are the research needs. And similarly, this is I think the risk and the safety and security components related to scale up are also important. The innovation component is also important here. Within that second grand research challenge, we broke it down into 4 key areas where there's a dire need for more research and more innovation. Within these 4 areas, I think chassis and process technology and equipment are likely most relevant to all of you, thinking through as we develop different chassis, what does that look like? What are the needs? How do we, even getting into some of the regulatory, how do we regulate and think about risk related to these? And then process technology and equipment is related to innovation for quality assurance, but then also innovation for, making sure that you can produce products quickly and effectively. So with all of that, I am also going to present on the regulatory component. I'm not a regulatory expert. We had Anastasia Bodner on the team, who many of you know, former USDA, and led the work on simplifying regulation for the commission. I am not her, but I'm happy to present her work, and I'm happy to talk about where the commission landed, where the whole commission landed.
Before we go into reg, should we just pause and see if folks have questions?
Oh, sure.
about
Yes, sorry.
the research challenge?
Let's pause and ask see if folks have
Also, so you can take a break.
Any questions about the first part before we launch into regs?
Just a quick one. In the recommendations, thinking about scale of concerns and, looking at kind of both keystone challenges of minimally synthetic cells and chassis. So were there conversations around particular chassis types that were highly recommended for a focus within this work, or was it pretty broad?
Yeah, it's a great question. It was pretty broad. We chose not to pick winners for like the conversations around all of the technical details around specific technologies. I think we just recognized the need for a broader range of organisms and also technologies to be able to pursue scale up, and the lack of, general research and funding in that area.
Go ahead.
I'll keep going. Okay,
We're saving them all.
no, that's totally fine. So, we spent a lot of time talking about, I just spent a lot of time talking about kind of the promote side of things, R&D and innovation. We're gonna touch on that quite a bit, but I want to transition over to talking about the regulatory space. And then I'm going to pass it over to Sam to talk about safety, security, and responsibility, because all of these components in some mix are going to be relevant for the report that you all are responsible for. So I don't think I need to tell this group too much about the state of the current regulatory system, but I will note that, of course, it's when it comes to biotechnology products, it's a bit of a patchwork, and there are a lot of challenges that are currently associated with it. So
Very coordinated patchwork.
coordinated patchwork, sure. And so one of the things that in pursuing the work on regulation for the commission, it ended up landing in kind of two buckets of things. And so as you are all aware, there's a huge number of challenges. And so some of these challenges can kind of be divided into two buckets. One bucket being kind of challenges with the current system and current products, and then challenges in thinking about novel and future products. So when we think about the current system, we think about kind of navigating overlapping jurisdictions, the lack of clear pathways for regulation, the lack of coordination. And then when we think about novel products, we think a lot about the inability or unwillingness related to innovation and adopting innovative practices for regulatory purposes. Of course, when we talk about kind of the patchwork and all, or the navigating jurisdictions, we're talking about the three regulatory agencies that are responsible for biotechnology: USDA, FDA, and EPA. And so as we kind of move forward, that's kind of the framing that we're thinking through. I want to note, and so related to those two buckets of challenges, we then have kind of two buckets of here's how we're going to solve things. So the first is the recommendation that you see that's 2.1a, which talks about simple pathways to market, especially related to familiar products and removing unnecessary regulation. And then the second is specifically on preparing for novel products coming to market. Of course, this is also built on the work from the Future Products of Biotechnology report and many others that are related. So we're really grateful for those resources as we kind of move forward in our own work. So I'm gonna move forward to talk a little bit about these two different recommendations and kind of how we see it fitting in with kind of the work that you're doing. This first one about simple pathways to market. This is very important and maybe a little less relevant to the exact work that you all are responsible for in this juncture. I will say that there is a lot of important aspects related to just making sure that products actually get to market. And there is a lot of need for coordination. So within the report and within our recommendations, the crux is sort of the key part of this set of recommendations is that we call for a coordination office at the level of the White House that serves a lot of these different functions. So works on creating these simple pathways, mapping these regulatory pathways, reducing unnecessary regulatory burdens, and working on kind of and all these other components that you see here listed. I will point specifically to the third bullet on conducting regulatory trials and inform clear regulatory pathways. We're proposing here sort of a regulatory sandbox model where the regulators and the innovators get to work together at the opportunities to understand what innovation could happen, and then use kind of the opportunities for quality and efficacy to make sure that the products, that the innovation that you're thinking about through the regulatory pathway is effective. I'm going to move forward a little bit to talk about our recommendations on novel products, but I'm happy to go back and discuss any of the bullets on the last slide further. But in the second bucket that we discussed, we're thinking about products for preparing for novel products to come to market. And so within this bucket, we have three components of the recommendation, and then I listed a fourth for infrastructure purposes. But the first one is related to bolstering regulatory agency capacity. We need people at the regulatory agencies. We listed some numbers in the report. I believe it's, there are 9 FDA workers who work on food safety, plants and microorganisms produced with biotechnology, and there are 6 EPA workers that are reviewing commercial use of certain microorganisms produced with biotechnology. That is, of course, at the time that the text was finalized, so unclear what it is right now. And so, without the resource capacity, it's very difficult because you need people in order to be able to move innovation, move research, move the actual regulatory process through. The second one here is establishing a foundation to enable biotechnology innovation. This is built on a model of foundations that's happened throughout the federal government. There's foundation at the national at NIH. There's legislation currently for a NIST foundation, and the model here being that academia, industry, government can all work together to come up with solutions for product regulation, and also to help resource and fund some of those opportunities where there are gaps in kind of the landscape. And the last one here is establishing last on the top set of bullets is the Federal Biotechnology Regulatory Research Program. There are research programs that exist. They're fairly small. The report calls for expanding and ensuring that they continue to make sure that we have the research capacity that we need in order to continue to move innovation forward in the regulatory pipeline. And the last thing I'll talk about here is actual infrastructure. So part of the report calls for centers for biotechnology at the DOE national laboratories. Within that recommendation, we specify that all of these centers should have a focus. And one of the foci is on test beds for risk assessment. And that directly relates both to the regulatory space, but there's also a really nice lead into safety, security, and bioresponsibility, because it fits kind of both of those categories and could serve both of those purposes. And so with that, I will kind of just simply pass it over to Sam.
I threw my background yesterday.
I do want to pause though for questions on this. Caitlin and I will.
Want to make sure I understand it. On the established foundation to enable biotechnology innovation, is that specific? It's underneath the regulatory part, right? So, but I guess also a little bit in the horizon scanning. So maybe can you say a little bit more about what types of things might you imagine a foundation could do or would do?
Yeah, I can start, and I'm gonna have Caitlin kind of jump in. I think it started specifically related to regulatory, and I think it's expanded a bit. The call for the foundation was actually to have it through the National Science Foundation within the report, because the idea being that the basic research, that's right. Oh no, that was the research program. Yeah, go ahead.
Just to say, the foundation is intended to be principally focused on research and support to the regulatory agencies. So it is not a foundation that is intended to do biotech innovation and writ large, but in service of, more streamlined regulation. And if I may, just to add one kind of big picture comment to tie up everything that Stephen said, the goal with both sets of recommendations, both the simple pathways for today's products, as well as preparing regulators to deal with the novel products of the future, is, the more time that our regulators spend on products that they know, that they are familiar with, that they understand how they work, the less time and frankly, bandwidth and you know, sort of mental energy they can spend on the novel products that we don't necessarily understand today, how they will work, how they will interact with their environment. And so the goal was to move, to streamline, to simplify, to, the pathways to market, the regulatory pathways to market for familiar products as much as possible. And as regulators become more familiar with new classes of products that are coming to market, that all gets kind of moved along so that we're spinning the regulatory flywheel in the same way that we're spinning the innovation flywheel. So the regulators are able to not just kind of keep up with what's on their plates today, but to, again, be more, future focused, more time and energy spent on staying abreast of the cutting edge science and being ready to quickly regulate the products that are not on the market today, but we know are coming.
And I think Melinda online had a question around maybe coordinating office or.
Yeah, I was wondering if there was any discussion of just scrapping the 3-agency regulatory approach. I know it'd be a pretty major change, but lots of countries have just one office of biotechnology regulation. And I could see that office being potentially staffed with individuals on detail from each of the regulatory agencies to meet the statutory requirements there. But was there any discussion of taking an approach like that as opposed to creating additional offices? Because I think a lot of this seems like it would require hiring a lot more federal staff and creating more offices, which I think in our current environment seems unlikely to happen.
Thank you for the question. I'll maybe address in 2 parts and do the second part first, which is our goal with the creation of the National Biotechnology Coordinating Office was that it would do far more than all balls and strikes between the existing regulatory bodies. We envisioned that it serves as the, very sort of muscular coordinator across the interagency for not just regs, but promote actions, protect actions that, what we diagnosed was that there was all kinds of really valuable biotechnology activity, both, in the promote side and on the protect side, but it is in a lot of cases, even offices or agencies within departments that had different aspects of the responsibility of each other. So we also designed it so that it could be fairly lean and nimble and not require a huge new, bureaucratic surge, particularly in EOP. So that just to sort of answer the second part, which is about, building something new in this environment. Your first question was, did we consider kind of reworking the 3-agency coordinated framework approach? And we did, and as I alluded to, I think you'll see us do more on regs over the next year. Please don't read that as like any indication of, exactly the direction that we would go. But what we really wanted to do with this first push on regulation was identify things that could happen right now that would meaningfully change the speed with which things can move through the regulatory products can move through the regulatory process and the complexity that developers face in navigating the interagency, coordinated framework process. And so it was a little bit of a tactical decision to put forward recommendations that would not require seismic legislative change. They do require some legislation, but as folks who track the regulatory system know, each of the regulators has their own governing statutes. In some cases, there's more than one. And so unpicking that starting from scratch is a much larger legislative task than what we put forward in this report, which was designed to be things that could happen, as early as right now, that would make a big difference, but more to come on more of the statutory
And I think we have a question from Felicia before we go to Sam.
Very, very quick, because I know we're running a bit short on time. Stephen, I was very glad to see that you gave that last bullet point about the interest in the Center for Biotechnology and Biotech Risk Assessment. There is a Society for Risk Analysis, a professional society. I'm its immediate past president. So we would certainly be interested in working with you all if that is a topic of interest. And to that end, I'm very curious as to what you all had in mind with respect to biotech risk assessment, because there's so many different areas. There's engineering risk assessment, human health risk assessment, and environmental and ecological. Do you have any particular foci for biotech risk assessment?
This is another case where I'm probably going to defer and say we didn't get too specific. I think we had been primarily focused on areas where there is a complete lack of infrastructure currently. So I think the environmental risk assessment comes to mind because there's minimal infrastructure there, but it's not necessarily that it wouldn't involve other types of risk assessment. We also are aware that there's lots of work, there's some work starting at various national labs related to different types of red teaming and risk assessment. A little different, but within the same vein. So that was kind of what geared us toward an interest in including some kind of center related to this.
Sam?
Great. So a lot of the recommendations that we've talked about so far have been really enabling biotechnology and enabling it to flourish. A lot of them through what is it that we need to do as a government that only a government can do that that creates this field for rapid innovation. I often describe my work on the commission as how could that go wrong? And so my bucket of things is a lot of my work falls into this category of protecting against the harms of biotechnology. Next slide. So our current system around this protection side has three general characteristics that characterize a lot of it. One is the tools that we have are fairly blunt and they're reactive. So think of moratorium, think of export controls, think of secrecy classification. Like these are hammers, sledgehammers, and when they come down, they realign entire industries. They're also reactive in nature. So something tends to happen, and then we do something about it, right? And one idea here is not having government do more than it needs to, but it also results, particularly in biotechnology, and things that having to happen that could have much bigger consequences than we're prepared for, because we're waiting for them to happen in order to get in place the governance processes that we would need to do something to prevent them from happening in the first place. Second point, the government leadership in this area is fragmented. So this goes back to the question online about like there being lots of different agencies involved. We think, contrary to the recommendations that we were talking about for regulatory coordination, we think in this space there is a value to understanding that fragmentation has led for no one agency to really see attention to biosecurity, biosafety, and responsible innovation as their thing, right? And this then leads it to be like a Saturday affair for them, right? You know, if they've got everything done in their week, they might have time and attention to turn to this. This isn't to say that there aren't a lot of really good people in the government working on these questions and in this space right now, right? I think what I'm trying to show though is that we don't have the priority structure from the top that says that this is the thing that we need to be focusing on. What this results in, these blunt, reactive, and fragmented approaches, means that our innovation in the way that we govern tends to be out of pace with the innovation on the science and technology of itself. And so as a result, these tools kind of wallow. There's a lot of there's an event, there's a lot of attention to safety and security or ethics. There's a panel convened like yours or others. There are recommendations developed. They form into a particular niche, and they are put into a government entity where it's expedient in the moment. And then we go on. Think gene synthesis security, right? We've been debating about how to do this for 20 years. We have we've had report after report. We've had committees working on it. We have guidance that finally got formed around 2013. And it took another 10 years for that guidance to be updated. That updated guidance is still voluntary, right? Or it's tied to federal funding. So there's a whole swath of the synthesis community that isn't necessarily directly covered by it. Think dual use research of concern and oversight policies in the space. Think about information hazards. If we go all the way back to the Mousebox papers in the early 2000s and the like, it's because it's nobody's concern directly, it's everybody's concern, right? But because it's everybody's concern, it doesn't actually get addressed systematically. So our recommendation overall is that Congress must direct the executive branch to advance safe, secure, and responsible biotech research and innovation. This is a broad recommendation. However, underneath it in our text in 4.4, if you go to the next page, we talked about the need that we've tried the fragmented approach. We've tried the reactive approach, and we've seen that it has come up lacking. So what we are arguing for is the need for a consolidated approach. This consolidated approach would have 5 key characteristics associated with it. First is a capability to identify risks and vulnerabilities at very early stages of the research process, and risks and vulnerabilities within the research system itself. So if you think about the regulatory pathway that we were talking about earlier as looking at the products and like following the product along, this is thinking about like, how does innovation happen? How does research happen? And what are the vectors within that process for us to be concerned about what research and innovation is done, who's doing it, for what purposes? This shift in mindset leads us to open up a whole landscape of opportunity here for innovation in security, safety, and responsibility. So think about this as an overlay of innovation with the research and innovation on biotechnology itself. It's innovation in laboratory processes. It's innovation in biosafety. It's innovation in organizational design for attending to the ethical and responsibility concerns of emerging biotechnology, right? And I'm mixing here the ethical, the security, the safety, the responsibility very purposefully, because these are all ways as a society that we become concerned about something, right? And sometimes it's like somebody's going to do something bad. Sometimes it's we're going to make a mistake, and sometimes it's like, is this a mistake, or is it a feature, or is it a bug, right? So these are all parts of the same coin, which my coin is now turning multi-dimensional. Apologize for that. But the point here is that the system to address them is similar, regardless of what kind of concern we're dealing with. So these 5 steps that we're going through here form, I'm going to bring this into focus on the next slide, but they form a feedback system. So first, identifying emerging risks and vulnerabilities. Second, taking those concerns that we've identified, whether it be from communities that are impacted by emerging biotechnology or through our own internal analysis or data streams of one sort or another, and funneling that into a research and innovation capability on biosecurity, biosafety, and responsibility. So actually new capabilities. Those then are also coupled with the development and incentivization of best practices in this space. And this presents a strong focus on the practice and process of research and innovation, and not just the things that are being innovated. Then consolidated and overseeing our policies and and you can see a selection of those up at the top. And finally, collaborating with the international community. How this works, how we envision a way for this to work in practice is on the next slide. So this is a proposal for, yeah, go for it. This is a proposal, that's way too small text for you, but that's okay, because you can see what we thought it through. The basic structure of how this could happen is if we think about an innovation engine here that includes within it a DARPA style move quickly and rapidly when we identify concerns that need to be addressed, to get the best and brightest minds to be working collaboratively with industry and academia to and the US government laboratories to rapidly design and test and be able to roll out technical and organizational ways of thinking about safety, security, and responsibility. Where do they get their ideas from? Part of it's like being at the leading edge of innovation in the space, but it's also listening. So this assessment shop is basically a listening arm. It's listening to those impacted. It's listening to the landscape and doing analysis on data flows from, for example, like a no-fault reporting system that on laboratory incidents. It is stress testing in a sandbox environment. So we have a sandbox here too, as a way of testing potential updates to oversight before they get rolled out. And it's stakeholder engagement. So it's creating a pathway that isn't just a Federal Register notice or a 5-minute public comment at a meeting, for you to be able to have a dialogue and engage and sustained interaction with a broad set of the community of people who are engaged in some way or another with emerging biotechnology and its concerns. That then, so this assessment shop feeds into the innovation engine, and the innovation and assessment shop together enable new options and new opportunities for oversight capabilities. So take the example of information hazards. We've been dealing with this problem for a very long time. It goes back to NSCEB 189, 1985, and even before then. How do we manage concerns about what we say and don't say about the possibilities of new technology? It's a hard nut to crack. Part of the reason it's a really hard nut to crack, and the reason that NSCEB 189 came out in 1985, is that there isn't a strong degree of trust between the academic community in particular and the government and industry on like what are your concerns, my concerns, and how do we match those up? That takes a sustained interaction to really achieve that. And we're proposing that a body like this could be a place to work that out over time. Okay. I think that gives you the basics of it. The oversight system itself, we're thinking of this like a version control process. So it's not a once every 10 years, when we get around to it, when there's a right moment to kind of let all of the stars align and then an event happens so that we can update our policies. We think of this as like, oh, this is version 6.2 of the census framework. Like, great, lock it in. This is what everybody has to do now. You don't like it, I understand that. We talked about that over here, but time to get to work on 6.3. So come on, let's get back to the research and innovation engine and figure out how to move forward. But right now, that's the policy. Also in this group is we're envisioning a lab licensing system, and that this entity itself would have civil enforcement capabilities for the policies and regulations it would oversee, but not criminal enforcement. That would stay with the FBI, other agencies. I think that's the main points.
Thank you all. So I think we have about 5 minutes for questions, but I noticed you guys cleverly built in a little buffer, so maybe we can squeeze just a little bit more time as well, because I'm sure there's some good questions here. And I will not ask mine first, unless one thing I will point out, anybody just please raise your hand. One thing I will point out though is you guys have about another 19 months or so. And so the timing also, as we're thinking about that for this committee and how what we're saying hopefully can be really helpful to you as part of that, and maybe will fit and be a nice nice timing as what, hopefully, as I said, what we can do would be useful.
Yeah.
So, that's what I think we're
Quickly on that, we recognize that, a lot of the problems that we propose recommendations for are things that this community has been thinking about and working on and making recommendations about for some time. We also know that, others are going to have different ideas about how to solve the same problems. And so what we have been telling our congressional audience and invite you guys to take a similar approach if it feels right to you, is to say, like, if you don't like the recommendation that we are putting forward, or you think EBRC's recommendation is better than NSCEB, is just to, take the two organizations, like, focus on the problem. Like, because the problems that we lay out in this report are common things that this community has been highlighting for a long time. Now let's work together using the time that we have left, using other entities that have a more enduring mandate, like the academies and others, to, again, if you don't like the solution that we recommend, let's work together on an alternative one, knowing that the problems are the right ones and the real ones. And, we can't not do something about them.
maybe following on that, just let's say, see some hands. You from the very beginning, you mentioned that you've kind of broadened your scope a bit in terms of not focusing just, I think it was not just on the like biosecurity pathogen space, but on the broader security implications. Can you just we're kind of thinking about that as well for how we interpret security or biosecurity or things. Can you just comment on that a little bit, and if and when or how much that might be helpful to you if you comment on that as well?
Sure. So one of the motivations for the scoping decision there was not to, there are places where we tread on other bodies, certainly, but, there has been a longstanding commission on biodefense. There are lots and lots of groups looking at questions of pandemic preparedness or COVID origins or any number of other related things. And so we really wanted to stay true to the mandate that was given to us by the Congress, which was not include those things, not get sucked into spaces that were already very well covered by other bodies. And so, in some places there's some overlap, but a lot of times when people think about the intersection of biotechnology and national security, their minds go directly to biodefense. And, that is important work, but there are other groups that are more focused on that. And so what we really wanted to focus on were biotechnology applications in other, in the defense sector for conventional military applications, not bioweapons, and then in other sectors that we felt, for example, like agricultural biotechnology or biotech for energy or supply chain resilience. Those were far less well explored by other.
Could I ask a question, because the community is sort of getting started in their work? Given all the work experiences, the conversations that you've had over the last 2+ years and what you're expecting in the future, what do you think the greatest value add and enduring sort of outcome this committee can offer?
That's such a good question. Thank you for a deeply unfair question. Look, this is part of why I said what I said to India, which is, we want to approach this with certainly the confidence in the 2 years of work that we put in this, and the true belief that we have stress tested these ideas to high heaven, and we believe that they are sound. So with humility, reinforcement that our ideas are brilliant by groups other than these 11 commissioners, extremely helpful. Additional stress testing of those ideas, right? And groups saying, we looked at the commission's ideas and thought that, A, B, and C were sound, but D, E, and F were off base, and this is why. Like, having a healthy debate around, again, either the ideas or the problems that underpin them. And then, we are lucky to have this, this 18-month tail, but we're going to close our doors at the end of next year. The academies will continue, you know, that these studies will continue. And so there are things that either we will want to be able to hand off, that we aren't, you know, either weren't able to complete or that need a champion. We did the work, we all did the work, but we need somebody to really, you know, carry the torch for these ideas if they're not implemented during our life, you know, the lifetime of this commission. Hopefully they'll be implemented during our lifetime, the lifetime of the commission. So those are all things that, you know, are enormously helpful to us. Reinforcement, debate and stress testing, or offering other ideas or other ways of thinking about these ideas. And then, you know, championing these things after the lifetime of the commission.
Richard and Felicia.
Sorry, Sam had one.
Oh,
additional thing.
We said this at the beginning, but we are a bipartisan commission. You all are a nonpartisan, you know, body. But we live in a world right now that has, you know, that has a lot of political forces, you know, running around in it. And so one of the things that we have tried to do in our report is to write the report for the moment that we're in. And maybe that's it. Maybe that's something for you all to think about as well. Like, what does it mean to write this report for the moment that we're in right now, in a way that allows you to maintain your position of nonpartisanship, but also like understanding that ideas taken up in the world require them to be understood and seen as their own by key actors in the space right now.
My question is a little bit of a follow-up on what Kavita asked, but maybe in a slightly different way. So maybe it teases out something different. As you were going through and looking at what's out there already and what people have talked about and what recommendations were there areas that you felt like, boy, this is an area where just there's not a lot of thought that's gone into it. There's not a National Academy's report on that particular issue. That's surprising. It would have been nice to see somebody have thought about that. But then it kind of came up as surprises in terms of how little thought, analysis, or other things sort of showed up.
The science of biosecurity and biosafety, like there isn't really a funding stream in the United States for attending to this. You know, we recommend that as part of this consolidated entity, but it is it's really surprising that it's seen as a compliance burden, and therefore as a thing that you create a policy for, and then it's an unfunded mandate to actually deploy that policy and to hold it up. Nobody knows how to hold it up because there's no systemic work on like how to actually do that well. You know, the Society of Risk Analysis, you know, it does this kind of thing in a more academic sense. The ABSA, the American Biosafety Association, does this, you know, in some of its training classes. But it's not at the level of like rapid attention and innovation and the literature and the field itself of how to do this that is tied to innovations that you see within the laboratories, within the scaling facilities.
Yes. Yeah. I think that's a really good one. I think one of the ones that struck me, and it may, I think it's relevant to this group as well, to all the groups I feel at least, is related to biological data. It's not, there's a ton of work on biological data. Don't get me wrong about that. But usually what you end up seeing is that they are diving into one very specific aspect and one key component and one thing that they care about the most. And it's very siloed and very, there's a lot of parameters around what the exact thing they're looking at is. And so when you talk about biological data more broadly, people tend to shy away from writing that information or putting that out into the world because you know, the first thing that they say is like, well, which biological data are you talking about? Which, like, what are you focusing on? What are you prioritizing in your own conversations? And so figuring out how to have a discussion about the landscape of biological data in the United States and, you know, conversations, we didn't specifically talk about prioritization, but talking about prioritization and talking about security and talking about some of these other pieces, it was difficult to find a ton of those. So
maybe to that, and Richard's point, I wonder if there's also some data, like say regulatory or others that might have been available to you all, but is generally not available to the public or to others that might also make it, just as some of the other comments we heard this morning about what's available in terms of regulatory information. So I think we have time for just one or two more questions.
Yeah, I've been wondering, I noticed in the language of the recommendations, there are many that use must and, many that use should and a few that use must.
Yeah.
I'm really curious as to whether there was a particular process or criteria that gets things to must.
Yeah. Process or criteria. The 20 somethings on my staff would call it a vibe, I think. No, we did have a process within the commission of, at the end of the day, we had more recommendations than are represented here. We had to make some difficult decisions. It's not only about, what we put out, but then what we, thought was absolutely essential and a must do versus a should do. And what we looked for was, against each of these problems that we identified, what is the thing that if implemented, even if it's harder to implement than some of the other recommendations or might take longer, would have the maximum impact in either running faster or slowing China down, depending on, which action we were working on. And so our goal was to focus congressional attention to the things that if they too were making hard choices about which of these 50, they're going to do all of these, obviously, like this year. But in case they weren't going to, that we would help direct attention to the things that we felt were absolutely essential, followed very closely behind by the must shoulds or the strong encouragement shoulds.
Yeah. Following on Pete's point, I think that I think it might have been you, Caitlin, we were talking about regulatory bottlenecks and how those maybe some of those fit into the must versus the should.
Yeah.
I'm very curious as to if you could describe what you see as being some of those regulatory bottlenecks. Does it have to do with a particular acts? Do we need a new act? Do we need what are some of the?
Sure. Stephen, feel free to jump in here, but I think that like a couple or maybe two that stand out to me. One is that when a product is regulated by multiple agencies, they have to navigate all of that largely on their own. A company looking for regulatory approval needs to navigate that largely on their own. And in many cases, each of the regulatory agencies that they interact with require different information in the submissions and the dossiers, different timelines for review. Sometimes, they go back and ask for additional information, and then the other regulatory agency kind of later, but in parallel, also asks, right? So you end up in kind of this eddy of, this group needs more information, and then this group says yes, but then the other group hasn't gotten to it yet. And so I think one of the observations from company, we did a lot of stakeholder. So one of the observations from companies was that moving towards something like a single window, single dossier, rather than making a company navigate this complexity, mapping that simple pathway so that you can say at the outset, okay, you are going to be regulated by all three because that's the nature of your product. But we're going to do the hard work behind the scenes of coordinating ourselves and making sure that we're speaking, as regulatory agencies with one voice back to product developers to say, here's your timeline, here's the information that we need, here's when you can expect to hear from us. And part of that is essential, not just for the product developer to make their own business decisions about, when are they going to be ready to scale? When are they going to be ready to put a product on the shelf? but say their technology or say their product is a component part of another technology, right? They're selling to a larger company. That helps them give certainty to their customers. It also helps them give certainty to their investors, and that has been a major issue. The other bottleneck that I would highlight is from the regulator's perspective, and I described this a little bit, but just to reinforce, the expectations that they have the same process for review of things that they have reviewed before, regulated before, that are familiar to them. We understand how that technology works. The getting use the word like sort of bogged down, but that's, I don't mean it as dismissively as that. It's just like having to keep, spending the same amount of time and energy on a product that you have seen, or analogous, product that you've seen a million times before, means that you are not spending time learning about things that you haven't seen before, truly novel products. And that creates a bottleneck also.
Thank you.
Can I do?
Really, really fast.
Okay, I just wanted to make a comment on the innovation side of things, because I don't know if I would call it a bottleneck as well, related to the idea that, we saw a lot of companies that were doing really innovative QA or efficacy trials with AI, with new instrumentation or sensing techniques, and they just weren't able to use those techniques or that process in the actual regulatory reporting, because it hadn't been approved. There hadn't been a way in which they could actually tell the regulatory agencies what they were doing, and it hadn't been compared to the current state of the art, which means that, even these companies that are really set on making sure that they're doing innovative things are struggling to get those to the market, which would then increase the speed at which products could be produced, be approved.
Great, thank you.
So we're out of time or over time. And so I really want to thank you all, not just for today, but for all the really hard and amazing work you've done over the last 2 years. And then also for today. We really appreciate you sharing this with us. And I think we will have a lot more questions continuing. So thank you so much. Everyone, thanks to everyone who's joining online. We're going to go in like 1 minute. We're going to have a very quick stretch break, and then we're going to go to our next session. So I'm going to back to Emily over there. Everybody can get up, jump around, and then we'll be right back. Yeah, please go ahead.
All right, thank you everyone for joining. I will be turning it over to our committee member, Peter Carr, who will be moderating this session. Thank you, Peter.
Hello, everyone, and welcome. We're looking forward to our next afternoon session, Artificial Intelligence, Automation, Convergence in Engineering Biology. My name is Peter Carr. I'm from RTX BVN Technologies, and I'm a member of the National Academies Committee on Environmental Safety and Security Evaluation of Engineering Biology. I want to thank the panelists who we're about to hear from, and thank those joining us online. Today, we'll be hearing from Mike Imperiali from the University of Michigan and Gigi Gronval from Johns Hopkins University. This session will explore the impact of AI and automation and their convergence with engineering biology on biosafety, biosecurity, and environmental considerations. It'll examine how AI and automation can both increase risk and enhance security and safety. Our discussion features panelists from the committees that authored the National Academies report from 2018, Biodefense in the Age of Synthetic Biology, and most recently, the Age of AI in the Life Sciences, Benefits and Biosecurity Considerations, which came out just recently this year. Now I'd like to turn it over to our panelists for their initial remarks. Mike, if we could start with you.
Great, thanks, Pete. So yeah, I'd like to talk to you today about the two reports that Pete just mentioned. I had the privilege of chairing the 2018 report and co-chairing the report that just came out a few weeks ago. So for the first study, we were tasked by DoD with developing a framework for assessing the risks associated with various applications of synthetic biology and then applying that framework to a series of capabilities. So for the framework, we defined 4 factors to consider: usability of the technology, usability of a weapon as a weapon, requirements of actors, and potential for mitigation. Within each factor, we developed a series of elements to consider. For example, in usability of the technology, there are ease of use, which gets to some of the issues that your committee is dealing with, rate of development, barriers to use, and synergies with other technologies. And each factor then had its own set of elements. In applying the framework, we were asked to assess current threats as well as threats that may arise in the future. We chose 12 capabilities to analyze, ranging from making an existing bacterium more dangerous to creating new pathogens and modifying the human genome. The study committee recognized that predicting the future, particularly in terms of when a capability might become realistic, is difficult. We therefore, defined a set of barriers and bottlenecks that currently existed at that time for each capability and recommended monitoring for progress that might widen bottlenecks and overcome barriers. We were also asked to determine how much of a threat each capability posed. Again, putting absolute numbers on each proved difficult, and we therefore ranked the capabilities from those that warrant a relatively high level of concern to those that warrant a relatively low level of concern, again, at that time. So it was a relative rankings rather than trying to give absolute assessments of what the threats might be. The new report was also sponsored by DoD, and it uses the 2018 report as a foundation. It's aimed at considering how artificial intelligence changes things, with a specific focus on pathogens that have the potential to cause epidemics or pandemics. The 2018 committee considered computational approaches as a bottleneck at the time that needed to be monitored in the future. This is an example of how predicting when something might become an issue is really not feasible because, as we know, AI has been advancing so rapidly. The new report also examines not only the risks associated with applying AI to biotechnology, but also the many potential benefits. This study was conducted on a very compressed time frame, but the committee still took a deep dive into 3 capabilities: design of molecules, modifying an existing pathogen, and creating a virus de novo. We framed the discussion in terms of the design, build, test, learn cycle. The committee determined that designing a single molecule is currently possible using AI, protein structure prediction, and design tools. This means someone could redesign an existing toxin using different amino acids, thereby potentially bypassing current DNA synthesis screening methods. However, it also means that AI can be used to design therapeutics and vaccines with which to fight against threats, albeit man-made or natural. The committee also found that using AI to modify an existing pathogen would currently have limited success. It might be possible to add a virulence function to a bacterium, for example, but whether it would work as predicted is not within the current capabilities of AI. Adding something to a virus would be even more difficult, as there are severe constraints on viral genomes that would present barriers at the design, build, and test phases. These build and test limitations, of course, could change with advances in applications of AI to laboratory automation. For similar reasons, the committee found that designing a virus from scratch is currently not possible. In addition to the same genomic constraints, there's a limited understanding of how a virus functions as a set of genes working together to replicate and cause disease. This is largely due to a lack of relevant data, which of course will be needed to train any AI model. These capabilities of AI will undoubtedly change, however, and to address this, the committee adopted a recommendation that's based on a similar concept as I just mentioned in the 2018 report. Specifically, the committee recommends an if-then approach with ongoing monitoring for advances in AI that would change its ability to produce dangerous pathogens and therefore trigger new ways of handling those threats as those capabilities came along. Finally, the committee recognized the tremendous potential of AI to be used to mitigate potential threats, both natural and man-made. Here, however, it noted that there must be investment in more robust data sets that can be used to train AI for these beneficial purposes. That's the end of my comments. I look forward to the discussion. Thanks.
Thank you, Mike, and we'll hold off on questions until Gigi has a chance to speak, and then also I'll keep an eye on the chat in the Zoom as well for folks who want to enter questions there. And Gigi, please.
Great. Thank you for the opportunity to talk to you today, and I'm sorry I couldn't be there in person. So I was a member of both of the committees that Mike chaired and co-chaired, and so I can talk about that. But I'm going to focus my remarks on another group, as my role of the co-chair of a committee that deals with these topics for BioMade, which I think Alta is part of your committee, and she was also the co-chair of the early days of this committee. So if you are not familiar, BioMade was created by the Department of Defense in October 2020. It's a manufacturing innovation institute, and it was designed to further U.S. competitiveness in biomanufacturing. This is important because biomanufacturing is poised to be one of the most important technologies of the 21st century and the source of jobs and economic security for the US. And one of the benefits of being a leader in biomanufacturing in this growing field is to be able to set the norms for how biomanufacturing will proceed, not only in the US, but around the world, as long as the US were to be the leader in that field. And so the committee that we started was inspired by previous work on these topics. As you know, the US has a long history of making sure that the ethical and legal and social issues are dealt with when it comes to advances in biotechnology, starting in 1990 when the US launched the Human Genome Project. But we felt that given that biomanufacturing was not focused on genetics or on medical issues, that it deserved, instead was dealing with novel bio-based chemicals, fuels, textiles, and food, that it deserved a fresh look, maybe different emphasis, and some new language to kick that off. And so we created what we called the 4S principles. They are safety, security, sustainability, and social responsibility. So I always mix up the order, but that's the order that's in the publication that we wrote about those about all of those. So safety is meant to encompass not only the controls and practices of the biomanufacturing, the work that goes into it, but all the work that goes into protecting people and the environment. Security is not only dealing with the potential theft of technology, but the deliberate misuse of it. Sustainability refers to not only the long-term viability of and how the biomanufacturing will interact with the environment, but also like supply chain issues and making sure that they remain viable for the economy. And then social responsibility is aimed to how biomanufacturing can produce good social outcomes. So those are the 4S principles. And so our committee defined these and published those, and those are incorporated into all of the BioMade projects that they fund. And related to this, we had a study that was led by a fantastic, soon-to-be defending DRPH student, Aurelia Atol-Jacua, who also works at RAND, where we talked to biotech executives, mostly from BioMade, but not exclusively, to see how they feel the 4S principles, the trajectory of those within their organization or within biomanufacturing more generally. We heard a lot about, why social responsibility, especially to the environment, was one of the big reasons why they got into this field. But we also heard the very clear message that if we want to see this vision of 4S, security, safety, sustainability, and social responsibility, if we want to see these principles realized within biomanufacturing, we need to ensure U.S. leadership to be able to put that into biomanufacturing, not only here, but to make that the international standard for global biomanufacturing. And mind you, this was an environment where we had U.S. leadership in biomanufacturing, but several peer competitors really, not too far behind. But I have to remark that in the last 93 days, we have seen a lot of degradation of U.S. leadership across lots of science and technology aspects. And so, we're stopping the training of new scientists. We're creating a lot of uncertainty that this is a good career path. We are canceling grants, canceling opportunities, politicizing science and even facts, and stopping a lot of critical research. So it is really, if the U.S. maintains leadership in biomanufacturing, it would be a big surprise that would be the case by the end of this year. I would suspect that China will be a very clear leader in science and technology if we do not reverse course. And even if we reverse course today, I think the amount of uncertainty and damage would still take a while to fix. So I strongly believe that we can reverse course, and I would love to see that these 4S principles, be integrated into the kind and similar types of principles be integrated into the kind of work that we see in the future. But at this point, I'm not sure that I can say that it's the U.S. that's going to lead in this.
Great. Thank you, Gigi, for those remarks. And I'm going to open up the floor for questioning.
Yes.
Oh, sorry. Yes.
Thanks for those presentations. Gigi, I just wanted to follow up with you. You said that the interviews of the executives, they really emphasized their commitment to sustainability and social responsibility. Were those interviews indicating that they thought that those principles were already built into the biomanufacturing industry, or that it was important to keep those principles as sort of front and center goals for how we organize what we do and what kinds of incentives and regulations we put on that emerging industry?
Yeah, I think many of it was kind of a given that was one of the main incentives for going into biomanufacturing, that they saw the opportunity to have a more environmentally friendly method of making the goods that we need. I mean, of course, money is a very powerful driver, and probably, who knows, at least it wasn't something we explicitly asked whether it would eclipse all other things. Most of the things that they talked about, and I'll put the link for the publication in the chat, but most of the things that we saw this as a potential lever to be able to introduce some of one of the principles that is not as well understood amongst the people we talk to, and that is about security. So there was a very little understanding generally amongst the people we talked to about what potential biosecurity threats there could be out there. They were focused mostly on IP, and they didn't really see any other potential risks. And even IP was kind of not as fully developed as I think that it should have been. So we saw this as maybe trying to maximize opportunities for sustainability and social responsibility could be an incentive for some of these other 4S principles.
I have a question based on something that Mike said, but which I mean, either of you could respond to. In the most recent report, the threat categories identified were like making a novel dangerous molecule, modifying something like a bacterium or other agent, and making something completely novel, like a novel virus. With the sort of AI enhancements, as well as contrasted with some of the limitations, I was wondering, what about the combinations of, say, a bacterium that's being modified to make one or more novel dangerous molecules? So essentially compounding a potential threat in one sense. I'm wondering if you care to offer a perspective or opinion on whether that becomes something that's of greater concern because of that combination, or of lesser concern because there's a piling up of bottlenecks as well.
Yeah, so that's a great question, Pete. So, because we were moving at such a fast pace on this report, we really didn't have a chance to go off into a lot of these little interesting questions like the one that you mentioned. I think my gut feeling is that, with each added component to the bacterium, there are going to be more and more selective and evolutionary pressures that might prevent that bacterium from being able to function normally. But again, as we collect more data that allows us to learn more about what works and what doesn't, then potentially those bottlenecks could be overcome.
One of the things that came out of this report is that we thought that, or we heard that AI will help to generate better ideas and to help in the beginning of designing experiments, but that you still need to do the experiment. You still need to, you can't, you have to see if that works. And we're not at a point where you can skip that step. So it may save you time in one place, but it's not going to help you at the hardest part, because ideas are not, they're not endangered in science. So we have plenty of ideas. It's a matter of being able to put them into practice.
Thank you. And Felicia has a question.
Well, thank you both, Gigi and Mike, for your great presentations. I'm Felicia Wu from Michigan State University. Mike, you mentioned specifically, and you both participated in this recent National Academies report related to the age of AI and life sciences. And Mike, you talked about not just looking at the risks, but also at the benefits. And when you were describing virus design de novo, I was wondering whether you happen to look at potential application areas. For example, the potential use of using AI to create viruses that are phages that can take the place of certain antibiotics, for example. Did you take a look at any of the beneficial aspects of using AI to create new viruses?
Hi, Felicia. Yeah, so actually, I don't think we really spoke about phages at all on the committee in any way. But the one thing I would note, and I think it applies to phages as well as eukaryotic viruses, right, is that the ability to even just put something together that's going to be self-replicating, much less cause disease, or in this case, maybe kill a bacterium. We're just not there yet. So I think the same sorts of considerations come into play. But yeah, that's really, that's a great beneficial use that we didn't discuss at all.
I've been reading a lot about phages lately for another project, and I think there's so much that we haven't yet done to catalog what phages we have and to go out and grab more and assess their host range. There's a lot of work to be done without AI that maybe AI can help with along the way to choosing clinical cocktails of them, but there's a lot of non-AI work to do.
Thanks.
Kavita.
Hi, could I ask a question about the 2018 report, the synthetic biology report? Did you all look at synthetic cells and or encapsulated synthetic cells?
I don't recall looking at that, but Gigi and Pete, you were there. What do you guys remember? I don't think that was the thing back then.
I'll switch hats momentarily. I do recall we had something not on synthetic cells exactly, but on the concept of synthetic de novo virus-like entities. There was an example from the literature at the time that we were using largely to indicate that, oh, something seems fanciful now, but hey, look, if you look at some recent research that's adjacent to that, there's meaningful progress that could point in that direction.
Yeah, so we have a chart in that 2018 report that might be useful where we kind of rank our order of concern of where things go, like what we were most concerned about. And what we were most concerned about was recreation of viruses. And our less concern at the moment was, recreating a bacterial cell that could be, pathogenic, and de novo, would be far beyond that. But we did have a chart that put that all of that together. And in the near term concerns, of course, were being able to create a virus that has a known effect that that could be reproduced in the lab, and that's certainly within current capabilities.
Can I ask a follow-up, please, to those, which is based on what you had done in terms of the analysis for both the synthetic biology and for the AI biosecurity one, what things did you come up with that you think would be most relevant for this committee as they think about safety, security, and environmental risks of, research involving synthetic cells and other particles, similar particles? And also, what things do you think were not discussed at all or were missing or were not available that you think would be particularly relevant to consider?
One concern that we had in our report, we just talk about a little bit, but we don't go into a huge amount of depth, but we say, like, when it comes to near-term problems, when how AI and bio could be misused, is the creation of toxins, novel toxins, and or novel drugs. That we didn't spend a lot of time going into that because that was not our charge. But we wanted to highlight that was a near-term potential, that problem that needs to be dealt with.
Yeah, I think I would add that I think, the framework from the 2018 report, I think, is pretty topic agnostic. So it theoretically could be applied here in terms of the sorts of things that this committee is considering. And I think also the if-then approach might be useful in terms of trying to sort out, whether there are things that are not possible now, but that need to be considered or watched for as the field evolves, and therefore just kind of keep monitoring things as we move along.
And then, since we were talking about the 2018 report, one question I was wondering was, I know that the focus of the recent report especially was the impacts of AI, but thinking expansively, whether it went into the recent report or not, I'm curious as to your thoughts on other things since way back in 2018, besides AI, that may or may not have there been other things that stand out, especially since that time, that have changed particularly in terms of technical innovations that would impact this landscape. You're free to say no.
No, I think, one of the things, right, that we sort of anticipated back in 2018 is the decreasing costs of DNA synthesis and the ability to potentially, just synthesize a large number of, molecules and kind of, throw them at the system and see what works. I think that, that's been moving along. So that's one thing, I think. And then I think also, we also, one of the other things we also discussed in the other report was, this convergence of nanotechnology and biology. And I think, of course, as is, evident from the COVID-19 vaccines, right? That's a real thing. I think that's something that needs to be considered.
So post 2018 report, that was when the synthesis of horsepox was announced and became a controversy. And we actually used the framework developed, and I can put the link in the chat. We used the framework in the 2018 report to analyze just how, and one of the useful things of that framework is like you have a new announcement, like, oh my God, you can make smallpox now. How much of an advance is this, you know? And so the framework allows an analyst to kind of stepwise say, well, okay, how does this really change our security vulnerabilities? And so we use that framework to walk through just how much that horsepox synthesis paper really changed our security vulnerabilities. And I think, you know, we came out a little bit like, you know, this was something that was possible before. This is, you know, now a proof of concept. It's not as easy as it may look on the surface to do this, but, you know, it is ultimately possible. And depending on which actor you're talking about, it is more or less possible. You know, I mean, if you have resources and talented staff, then it's certainly going to be more straightforward than if you're trying to figure out how to do basic things at the same time as doing some advanced stuff.
Yeah, and since we're mentioning DNA synthesis and large scale construction, it reminds me too that I think since 2018 in 2018, people were talking more about benchtop synthesizers, and there were certainly long existing oligo synthesizers from way back, but some of the more recent advances in benchtop DNA synthesis have happened since that time, it occurs to me.
Mm-hmm. Yeah, I think well, there's also been more emphasis on controls for DNA synthesis generally, and I don't think that there was a lot of concern about powerful bench tops synthesis that hasn't really taken off commercially. So I think that, you know, it's still. I don't think that it's changed all that much since in the last couple of years. But happy to be corrected.
I'll point out too that Gigi and Mike have put some helpful links in the Zoom chat for those interested. We have other questions. All right, well, at that point, I'll just ask Mike and Gigi if you had any kind of closing statements, post-discussion, if there's anything you'd care to add. We're certainly open to hearing.
Yeah, I guess I would just, you know, come back to one point I made very briefly in my opening remarks, which is that, you know, the applications of AI to automation of laboratories are going to potentially change a lot of the things that can't be done. And so that's certainly something that I think, you know, the committee talked about quite a bit, you know, kind of getting, as Gigi mentioned, getting from that design phase to the build and test phase. Once AI starts impacting build and test, that's going to change the equation quite a bit.
One of the people that spoke to our committee described AI right now as being like a great graduate student, you know, or a graduate student. Like sometimes some suggestions are better than others, and you know, they all still need to be evaluated. So I think that there's definitely hopefully it will get better. I just would like people to remember that there's positive aspects of these technologies, and you know, sometimes I think we get caught up in some nearer term risks. But you know, the benefits of them are could be really life-changing. I just would like them to be life-changing for people in the United States, as opposed to the places where I fear that are going to, you know, the places where science will be funded and performed at the highest level.
Great. Well, I want to thank both of you for joining us and sharing your insights with us this afternoon. It's been great to have you here. Thank you.
Thank you.
Thank you, Pete. Thanks, everyone.
All right, thank you for joining this open session. I will turn it over to our co-chair, Felicia, to moderate the session. Thank you, Felicia.
Thank you, Emily. Welcome, everyone. We're excited for today's session titled Open Discussion Between the Committee and the National Science Foundation. My name is Felicia Wu, and I'm the John Hanna Professor of Food Safety, Toxicology, and Risk Assessment at Michigan State University, and a co-chair of the National Academy's Committee on the Environment, Safety, Security Evaluation on Engineering Biology. So today we'll be hearing from Dr. Teresa Good of NSF, and in this session, we'll be hearing perspectives from NSF regarding the committee's statement of task. So, Teresa, we thank you for your time today. Following some remarks, we'll be engaging in a roundtable discussion with the committee to ask questions about the statement of task. So now I would like to turn it over to Dr. Teresa Good.
Thank you, Felicia. Thank you all. Thank all of you for participating in this really important work. This work was NSF was charged to enter into this contract with the National Academies as part of the Chips and Science Act of 2022. And the scope of it was initially very broad. It was all of engineering biology. It was all environmental, ethical, legal, social, societal implications. I think we had a great interest in equity and in lots of issues around the practice of engineering biology and how we could ensure that it was that this technology left the lab, went into, kind of was translated and had impact on everyone and benefited everyone. And I think that while that is still a really important goal, given the kind of need for kind of fiscal responsibility, I think that we have been asked to kind of reduce the scope so that we can ensure that we still deliver to US taxpayers kind of a benefit from the study that will enable us to ensure that this important technology that can get translated and can have impact. But we are we have to reduce the scope to ensure that we comply with some of the cost cutting measures that we and to be fiscally responsible to the taxpayers. And so what we have asked is that we really focus on things that haven't been done before, the things that we think will be most important for us to be able to ensure that we can safely apply parts of engineering biology, not all of engineering biology. We're going to focus it on synthetic cells. We're going to leave out proteins. We're going to leave out other things. Focus on cells, collections of cells, anything. But that was let's narrow that down and let us focus on those things that will enable us to ensure that we can safely translate this technology so it benefits the taxpayer. It benefits all. And so that is part of the rationale for what we chose in terms of the kind of how we rescoped. I think we had some conversations with the National Academy about, so what has been done before, what has not been done before, how can we ensure that we will have impact in the time that, and what we can do in this study. And so that's, I think, our starting point. And really, as I think about, we ran this solicitation for many years. It was called the Designing Synthetic Cells Beyond the Bounds of Evolution. It was my favorite solicitation at NSF. People would come up with ideas to create new things that we had never seen before. And they could have great applications. Some of it is just, what can biology do that we didn't know about? And then there's, well, then how can you use that? But in creating those new organisms that we had never seen before, I think that there has to be a framework by which we look at those and go, oh, this one we shouldn't do. This one is just maybe too risky. And as the kind of, or this one, there could be lots of potential applications. And I think that having a framework to look at synthetic cells in that way would be very useful for all of the federal government, and hopefully will profit us to be able to apply more of the technology that we develop. And I think it is both important if we think about what is contained and what is actually going to be released into the environment, because many of the applications will actually be more useful if they are in the environment, that whether they are, kind of addressing problems associated with reclaiming critical minerals or doing other things, they're going to be environmental applications, and anything that is in the laboratory could potentially escape. So we really have to. And so that is also why we specifically said both of those things. And so that was our starting point. And as Scouts, I believe the expertise in this room is far greater than, Kavita and I are smart, but the group of you are far smarter than that. And there may be some, what I really want, what I hope you come up with are recommendations that will help us proceed in what we hopefully will support over the next decade. It will, but we'll have a rubric by which to frame those decisions so that we can ensure that what we do has the potential to safely have impact, and be accepted, all of that. So that's my starting point. So back to you, Chair.
Wonderful. Thank you so much for that, Teresa. Now I'd like to open it up to the committee. We had the chance to discuss the new statement of task earlier today, and many of you brought up many interesting questions and points. So if you'd like to start to ask now, please feel free to do so. Yes, Jason.
I can start. I'm Jason Delporn from University of Wisconsin-Madison. One of the discussions that we've had, we understand the scope is narrower to synthetic cells, but we're also thinking about how big is the field of synthetic cells? And one of the questions is, do you want us, is the vision for us to focus on what is very unique about synthetic cells compared to other domains of engineering biology? Or is the task to focus on the breadth of synthetic cell work that actually overlaps with a lot of what is already going on with engineering biology? Does that also make sense?
I'm thinking about, I maybe get it, but I'm digesting it. So that as I think about what is a synthetic cell, I think that if it is something that is very, very close to E. coli, but I just made a tweak, that I think I have a rubric and I understand how to safely implement. I think the further I get from something that is not naturally found, that then it gets to be harder and harder, and I have no guidance for that. And so it isn't so much that I am interested in any particular type of synthetic cell. I am simply the further it gets from my lab strain of E. coli, then the harder it is for us to look at, is this change so much so that I now need to think about it really differently? And so that is kind of how, I don't think I answered your question, but I gave you my, hopefully that helps with your thinking about what you're asking, which is still gray and broad.
Great, thank you. Any other questions from the committee?
Kind of picking up on that. In the statement of task, it says synthetic cell, and you said not protein, not these other pieces, but maybe cells or collections of cells. And so we were trying to think about exactly what is meant by, or the scope of synthetic cells and how narrow, because if it had said a fully synthetic cell, that might mean one thing, or a minimally synthetic cell or another. But yeah, again, the idea that we can look at those chassis, think of it in terms of the chassis and the circuits and things that might be put in them, and those both would be parts of synthetic cells, versus, again, like the really minimal definition of it needs to be a fully living synthetic cell or something. And so I guess that's where we are just trying to think about where is it centered versus how, again, broadly cell-free systems and other kinds of things that are very tied to that.
So if I think about going to EPA and asking them, I have this E. coli that's got a new circuit in it, I think that EPA will know what to do with that, I hope, right? But if I come with Well, I've changed the membrane a lot, so it's got the, but I, and it's no longer a phospholipid membrane, it's something else. The further it gets from, what a known naturally occurring organism, then, and I think that it is a spectrum. And so I am not gonna be comfortable for me saying it's gotta be everything synthetic, because I think that is too narrow. I think there's a lot of gray of what we do, but it's very close to the naturally occurring organism. I know what to do if it is one gene, a couple of genes, but there gets to be a point, and if part of your deliberations is, what is that tipping point when it is no longer easy to use the old rubric? And we have rubrics for if it's a pathogen, right? But when did I make something where I no longer know it's not enough, it's not good. It has changed character, and it is it's spectrum, and I am not like traditional. Yeah, and I think we have a lot of experience with what I would say traditional metabolic engineering. But I think the further and perhaps certain circuits we're pretty comfortable with, but the further we get, the more we change, the more that it's not just a few genes, but it is that we're changing organelles, and we're changing ribosomes, and we're changing the nature of the polymers that organism is making, the further it gets from something I knew, and I know exactly what to do. So I am really.
So, Emma, you can go ahead, and then I have two questions.
So this is maybe thinking about that a little bit. And again, another way we've been trying to play with and understand the scope is thinking about, is it, the sort of the technology of the synthetic cell, but things like function and application of those cells that we should also be sort of thinking about, and how that relates. I think some of it, I think you were getting into that in a way with what you were just saying.
Right, and I do think that it is that the more we change, the more synthetic components we put in, then the more the function is less predictable, or that I can compare it to what was the host organism, or the original chassis, unadulterated chassis. I am not being very precise with my words this afternoon. Anyway, so I think it is in part, it is, what are those? Have I created new functionality that I didn't have before, that is perhaps not as predictable based on comparing it to the original host? And then I think that there are questions about whether it is that function contained, and then I think we all kind of believe that nothing really stays fully contained. But my graduate students certainly were not great at, things left the laboratory. And then what happens if it's not contained? And something, the application will surely be that we wouldn't have wanted it to be changed, but we're hopefully getting closer to answering your question, but I'm still not being great. Okay.
So when we were having these discussions earlier today, we came up with two questions that are somewhat related. One has to do with this, in the statement of task, the focus on biosecurity. And we wanted some elaboration from you as to whether indeed there is to be to focus on biosecurity, or whether we could consider other aspects of security, such as economic security. And related to that, we were interested in understanding whether, in addition to assessing the risks associated with synthetic cells, we ought then to also be considering the benefits, as well as the risks of non-adoption.
That is a very good question. And I do think that as we So some of the words in here, I believe my colleague wrote while I was on sick leave, but so perhaps but anyway, so take that, Charlie, I hope you're not listening. So I believe that it would be a missed opportunity if you only focused on the risk and not on the benefit. And as you develop whatever rubrics you develop to think about this and to help, kind of the agencies, the public, think about what should be adopted. If you do not also think about the benefits, then this would be it's a missed opportunity. So yes, do that. That was we missed that in the ways that we put together the new statement of work. I think that I would also want you to think about all of the aspects. I do think economic security, national security, all of those are appropriate things that you are thinking about. Risk and benefit, that there are potential huge economic consequences of not adopting. And I think that has to be part of the considerations in whatever we do.
Thank you so much. That provides us with a lot of clarification. And I believe Robin Scheffler online has some questions as well. Robin?
Yes. Thank you. And actually, this is exactly the question I was going to, so I guess, continue discussion on. A number of speakers that we saw today during the open sessions mentioned sort of weighing risks and benefits, questions of there's a lot of good to be done from these particular technologies. But I guess the question I have in terms of understanding the scope of what we're being asked to do is that if you actually look up words like safety or biosecurity, they're typically defined much more narrowly. And there is a bit of gear grinding here in terms of the types of hazards that are typically associated with, biosafety procedures, and say the long-term benefits, which might be, decades or generational of economic returns. And so those suggest very different ways of framing safety, back to sort of a, FDR style, sort of what is freedom type of a historical discussion. Like, then I'm just sort of highlighting that as a challenge in terms of how we might understand safety or security based on economic growth and its potential for positive benefit on a community versus the more narrow sense of biosafety and biosecurity that we've seen in some of the other documents.
Great. That I think that I wouldn't want you to simply do something that was so narrowly focused that it duplicated what we have done with DIRC or PEP or things like that. That would be you will simply spend a lot of time duplicating something that's already been done, and that is not your time is too valuable for us to do that. And there is a moment in time where we think we can still have an impact. In terms of our intent is not to have you focus it but stick to those narrow definitions. In terms of the timeline for, say, economic impact, that is a much harder question. That's a harder question for me to answer. And I am I'm going to struggle with that one, and I will try to get back to you with an answer, where it is reasonable. And in your deliberations, I would hope that you'll land on something that makes some sense, even if I fail to give you an answer today, which I apologize. I'm going to do the best I can to get back to you about that.
Great, thank you for that. Any other questions? If not, I will highlight something that actually Robin had brought up earlier today, and this has to do with recommendations that we would be giving as a result of our report, that we might make recommendations for NSF, for NSC, for OSTP, and yet a lot of the risks and the benefits would be realized at a local scale or at an institutional scale. So can you give us some insight as to kind of a balance of different types of recommendations to different stakeholder groups that you would like to see?
So part of why we said that it would help federal agencies and NSC or OSTP is as a funder of that sort of research and as the kind of policymakers around some of what happens in that space. I think that certainly for us at that level, I think what we're looking for is when I bring, if I convince Richard to ever become a rotator at NSF and he would run programs in, in synthetic biology, I want to have for him and others have a framework by which to think about how are we making those decisions. And I think that when Congress comes up with, I think that when Senator Rand Paul has reintroduced the Risky Research Act, and as we think about what sorts of things might be risky, how do we evaluate any of these things, and how do what of this portfolio should we be sending for their review? So there is on that higher level. I think that as local communities, if I'm gonna if my local community is gonna do they're gonna have to get permits to release organisms into the environment. But if they're going to consider that this is a way to, I don't know, remediate soil or do whatever that, or sequester carbon, whatever that is they're trying to do, that we would like for them to have a way to both speak to the public and to make decisions. As we have this engineering research, which is the Science and Technology Center that we fund at Cornell, that is the Center for Research on Programmable Plants. And they think about what kinds of changes they're going to put in corn and tomatoes and big sweet potatoes. And most of them, they have started thinking about things related to using less water and or adapting to drought. But and then they ask the kind of farmers how they feel about that. And the farmers are more worried about how much fertilizer they're using. And so I think that the risk and benefit at that level are very different than the kinds of risks and benefits or recommendations that I want for a funding agency, right? And so so if you can think about it as what would be appropriate for, the Risky Research Act and for, at the federal level as we develop policy, but what also would be appropriate at the institution or project level as they're thinking about what are the technologies that they want to develop and that will benefit their communities. If you can, but at some point, computer's going to go, we didn't ask you for enough money to do all of that.
Yeah. You did just mention the Risky Research Act.
Okay.
Yes, and I think we as a committee would be interested in understanding your thoughts with regards to that act as it may or may not pertain to our.
So it's not, there are lots of bills that get introduced and some of them they just get introduced over and over again, and some of them actually get passed, right? So I don't know about what will happen with this one, but I do think that there's a desire on some level to have more broad oversight of certain classes of particularly risky research. And I think that, but as we, like, and if it's strictly a pathogen and gain of function, I think we all kind of know how to, what belongs in that category and how to deal with it. But the further we get from the more we use synthetic biology or engineering biology, the more we use AI, but we will get further and further away from how we know how to precisely define what risky research is. And so I think that will become a little more challenging for implementation. What should go before a committee who has the expertise to actually look at that and determine do the benefits outweigh the risks? Are the risks mitigated? And so I think that, and so if there is a framework by which to think about that, that will be really helpful in the future. And again, I have no particular opinion about whether or not it will pass or if it should pass. That's not my place. But if it does pass, there'll be some challenges with new technology. Thank you.
Making me think a little bit about sort of how present versus future oriented this is. So just thinking about the kinds of research projects, for example, that are crossing NSCEB's desk now, and whether this is, a framework that is immediately applicable. You're saying, oh, the further and further away we get from kind of known things. So it'd be great to have a little bit of a sense from you about kind of where we are in that. If it, like, does it feel like there are projects now? I'm not asking to name projects, but that sense of like, is this something that we're trying to? Are we working several years, kind of trying to anticipate what's coming down the line several years from now, grounded in directions?
So I think there is tremendous creativity out there in the community right now. If there were sufficient funding, there would be. There's a lot of things that are happening now, I think that, and whether they're crossing our desk or a desk at DARPA or there's when I first started funding synthetic biology, maybe almost 15 years ago. It's a really different field. And the capabilities of what we can do is really different in the ways we are trying to, and so, it's different now, and we don't have new rubrics, but we still look at everything kind of through the lens of, does it look like a pathogen?
Thank you. Any other questions?
Got a couple.
Okay.
So let me maybe summarize a little bit of what I took away, and then you can tell me what parts I missed, if any. But then I have a question. Maybe I'll try. I don't know. So it sounds to me like a statement might be synthetic cells are an emerging area of engineering biology for which there's been less attention by regulatory agencies, national security agencies, and other parts of government. So that implies there's an opportunity to provide a framework for thinking about biosafety and biosecurity that could guide decisions as the technology evolves.
Yeah.
Maybe a little bit more specifically, I think consistent with that it would be useful for the committee to work to articulate both benefits and risks, as well as possible frameworks to assess and quantify those risks and benefits in areas related to synthetic cells, especially where there are new elements in synthetic cells that require changes to the existing frameworks compared to what exists for other parts. Reasonable statement. One thing I guess I would, building on that and kind of focusing on the synthetic cells, something that's new, both through the designing synthetic cells beyond bounds of evolution and the earlier understanding synthetic cells programs that NSF has run. Do you see specific things coming out of those programs that, boy, the committee should make sure to look into this area because it's an example of something in synthetic cells. I mean, you've given us a couple, but something like, well, here's something that surprised you, let's say as a biologist, right? That goes beyond evolution or is different than, right? Anything that comes up, just to kind of, make sure to look at type stuff.
Right, well, and, I think that when we ran that first ideas lab on synthetic cells, I think that there was a, project to build cell models out of carbohydrates and a project to do a mirror cell project with many ideas that are touched. There were some, different things around using excitability in cells. And I'm sure there were some more of them. And when we first looked at that, we were just like, this is cool. We will learn a lot of biology about it. And that was the end. This is cool. We will learn a lot of biology. We will learn what works and what doesn't work. And yeah, there could be some applications, and this is great. And 10 years later, apparently that was maybe not the best, but we should have thought a bit more broadly about that. It's not that we shouldn't have done it, but I think that there's, I think that there could have been, and so I think, do I worry about when people are using condensates to do all sorts of control of, cellular function or trying to create new organelles? Yeah, I don't really worry about that. But I didn't worry about so I am not, Richard, the right person to ask about what to worry about because I am still a scientist that thinks, everything's cool. And so that is why we need smarter people than me to come up with a framework.
But that gives some ideas, I think.
Right. And, I think that, everything out of RNA, again, don't really worry about that. But everything, backbones, again, I don't really worry about it, but the further it gets from something that the immune system could identify, well, maybe I should worry about it more. Anyway, so. Yeah.
I mean, I like your summary statement that you've made now multiple times, right? The further away that things get from things that we experience in nature, the harder it is to apply, right, the existing frameworks that we have, because they're often based on relatively small changes from what we see in nature. We're going to make a couple of small genetic changes, but really the cell's still living. It's going to propagate the same way. It's going to do horizontal gene transfer the same way, all those things. We've been, regulating that for decades, right? So we have a lot of experience there versus, something that might be really quite different. So I like that as a way to think about it.
And you'll be you like to ask a question.
I mean, yeah. I actually have a clarifying question and then a normal question. So let me ask my normal question first, which is, I recall very well that during the active evaluations of gain of function, we had to deal with risk and benefit, and the risks were often quantifiable and the benefits were often qualitative.
Right.
And it was really hard to sort of compare and assess those together. And in this effort that Felicia had mentioned in terms of thinking about the benefits as well as reducing those risks, how do you view the quantitative, qualitative aspects of those assessments?
Doctor.
So like some of them, like as I think about benefits, some of them are kind of, these are what we think of as good or whatever, that we're doing something that will benefit like if we did something that mitigated climate change, well, there's no, you cannot, it would be hard to put a dollar value on that, but we could think that down the, we can imagine what happens if we don't do that sort of thing. And so I think that it's kind of a fuzzy, but yes, I think that it does it. Yes, that's what it's going to look like. And there is, and in some cases, you'll be able to be more quantitative about what the benefits are. In some cases, you're gonna, the benefit is partly, yeah, it's a good. And if you don't produce that benefit, then the consequences could be, there are consequences not to.
Yeah, that's really helpful. Just to put into context, during that whole gain of function discussion, it was easy to quantify the risks because they were looking at public health, outcomes, whereas benefits, people thought that it was harder to be able to achieve the benefits, so it became a very qualitative exercise. So maybe that's something to think about as you're sort of thinking how to manage some of those things. The clarifying question, economic security, what I heard was, and tell me if I'm right or wrong, the far, economic growth or, the ability to commercialize something is well outside of the scope of this, but things like, considerations of how you protect or fill vulnerabilities for data sets and, algorithms and other sorts of things, research security and other sorts of things that might fall into that more tangible, immediate types, that would be potentially part of consideration.
Sure. I think that it's that in the time you have the amount of what you can do, then yeah, you can have the limit of how far you go. And so that is a reasonable way to parse it. And I will go back and think a little bit more about that and make sure that we.
If I could follow on to Kavita's question, sometimes what we do in economics is that when we're attempting to assess benefits, when it is difficult to come up with really precise and accurate numbers, is we imagine a best case scenario, a worst case scenario, and some probable scenarios. Is that something that you would welcome for this particular?
Yeah, would be with is, to get my training. It's not necessarily the way I think about it.
Wonderful, thank you. And I see that David Dunning has jumped in, and I wonder if you have a question for Teresa.
Oh, no, I literally just jumped in, so I haven't even had time to formulate a question. So I will attempt to do so in the remaining minutes.
Wonderful, thank you. Any other questions? Oh, yes.
Oh, something, Teresa, hi, Teresa, Pete Carr, I'm at these days.
You from 2010. Of my time in this.
One of the things you mentioned about kind of the early days of synthetic cell brainstorming, like a lot of the rationale being, oh, it's cool and we'll learn a lot.
Right.
And that's definitely a powerful motivator. Some of the stuff that we've read most recently still seems to focus on that rationale, not everything. It's cool and we'll learn a lot. And so I was wondering, and I'm a big fan of those, but I was wondering if, from NSF's perspective, if there are any particular application areas, or without being constraining, say, example application areas, things that have come across your desk that you might point to that would be interesting for us to focus on or consider.
Yeah, so again, we are, as the National Science Foundation, we mostly do that any discovery may eventually lead to that innovation that's gonna, change the world. And so there is still a lot of it's cool, we should do it. And we're gonna learn a lot. and there are lots of applications that are potential applications in really thinking about kind of food production and thinking about climate change and thinking about supply chains, thinking about ideas, other kind of critical minerals. I think that, and a lot of those are environmental. And I think that those ones are particularly, interesting and challenging. I think that some of the whole discovery stuff is going to have, I think, probably the first applications in medicine. And I think that those are, again, and our willingness to accept the risk benefit calculation always gets to be a little different than, I'm willing to take all sorts of risks with you. That including Alzheimer's disease. Anyway, but anyway, so I do think there are lots of new things and that we may be willing to do them if they're in. And I think, look, as you think about kind of that risk benefit calculation and how different it is when it is for something like food production versus if it is for medicine, and the economics of it, they really will change a lot. So it'll be interesting. I don't know if Kavita's going to give me the look to say it's getting outside the scope. You're asking them to do too much, but.
Thank you. I appreciate hearing.
Tricia, I just had a quick question. This is going back to something that the committee discussed sort of early before the scope, so it might not be relevant anymore, and Kavita might also give me a look for expanding the scope, but everyone looks anything. So I wanted to know, because you mentioned about having a rubric that NSF can use to assess something that comes across your desk. So thinking about like at the proposal stage or the early phases of getting something started and ideation and all that, I wonder, are you also interested in looking at different TRLs, the technology readiness levels? And then given that biomanufacturing seems to be pretty relevant in this case, looking at, MRLs as well.
Wait, as I kind of think about this, I think that the kind of, I suppose that the risk does change depending on the quantity of whatever organism it is or synthetic organism you've got. But the properly inherent risk and benefit, I could assess even at the very early discovery stage. And that, that I suppose if it is only a risk because I'm going to produce, and it might ask to, the Chesapeake Bay or something, then I suppose that's something to think about. But I think the inherent risk, you'd be able to tell even without thinking about the higher manufacturing levels. Sure, but that is just my framing of the world from I come from NSCEB. So, and I'm currently in the biological sciences, not even in engineering anymore. So,
really poorly formed question. So
then I promise to do your part.
Hey, I'm trying to figure out exactly how to phrase this. So one of the issues when we're thinking about the risk or security or those kind of, and even the assessing and mitigating these it's biosecurity, biosafety, environment. Sometimes if you think about how to do that, you could come up with sort of like approaches or a research agenda or investments that are really focused on the mitigating side or even on the defensive side. And so that ends up driving the research versus questions where if you're approaching it from what is already happening or what are the benefits that can or should be happening that would be driving the research. And if that happened, then what should the maybe mitigation or assessment component be? And so I guess I'm trying to figure out how to balance, like, how much should we be thinking about what's needed to mitigate as sort of the driving force as compared to integrating into whatever a research agenda could or should be. I tried to make that sound like a question.
Right, no, and I'd hate for you to spend a lot of time on a lot of different case studies where you go through lots of detail of this is how I would mitigate all of the risks for that, because I don't think that that will give me a broad generalizable framework to think about how I evaluate. Because hopefully there will be new innovations coming all the time that I'm going to have to, that we will all look at, or communities will look at, whatever, and we will have to look at a framework and then be able to apply that framework. And then from that framework, think about how to proceed. And so I would be much more interested in what would be the principles by which you would think about it, not specific examples of mitigation. Does that make sense?
Yeah, and I think it fits really nicely with your question about the, like the framing you use, or the kind of high level questions you might ask, or the approaches that might be needed at a foundational research or initial kind of thing, versus as it becomes like real, like what are the kinds of questions and how early are to involve those versus other. So maybe that kind of tracks a little bit with the TRL level series or something. Yeah, thank you.
Fantastic. Any more questions from the room or from online?
Maybe just to head one off. I think it's fine. I mean, I think we have room in here, but when it says specifically, there's like with NSF as our audience, or who we're making recommendations to, NSC and OSTP. There's some of these when we're thinking about assessing or mitigating. Some of these other things seem very much measures and standards and NIST potentially involved, or other agencies.
So that was the and other agencies.
And other agencies. Okay, good.
Right, that was
yeah. Okay. Oh, yeah, as well as federal. Okay, yeah, it's in there. That's good.
And that it is really, it should be very minor player in that, and we will have it will be very minor player in the grants.
Sorry, can I ask one more question? You might have asked the others this when you talked with them, but from your perspective, how do you think what would you do with the outcomes of this work? Like what are you thinking about in terms of next steps, recognizing that no answers have been given quite yet?
So I would expect that, given kind of the broad interest in biotechnology at OSTP and in Congress, that this is still considered one of the critical emerging technologies, and that synthetic or engineering biology is a driving piece of that, or that is the intersection of AI. But it is, and so it will be really important. So this is going to be something that we as are going to continue to be trying to enable that innovation, whether we do it through, whatever mechanism. So we will do that at a federal level and through public-private partnerships. And so I think that our audience is, that this should be a benefit to the entire ecosystem that supports that innovation ecosystem in the area. So that's my goal, is that I hope that you not only do you report out to us, but you take the show on the road, and you talk to Congress, you talk to the other agencies, you talk to STP, that it is broadly disseminated and that it will be a useful study in terms of if this risky research act is passed, then you will be the framework with which they really think about what of the things we don't know about pathogens, what do we really need to think about? And you will be the way that they think. At the stage.
Any other questions for Dr. Good? Or online? Well, if not, thank you so much for your time, Dr. Good. We really appreciate it.
Well, thank you so much for having me. I really do. I cannot thank you enough for all of the effort, the intellectual power you're all going to put into this and the work that you're going to do. That will, I believe, help us really continue to move this technology forward. So thank you very much for everything.
All right, thank you.